Hello, everyone, and good morning, good afternoon, or good evening, depending on what part of the world you are joining us from. My name is Nicole Kleinstroyer, and I am the Executive Director of the Interagency Coordinating Committee for the Validation of Alternative Methods, or ICCVAM, and the Director of the National Toxicology Program's Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM. And it is my great pleasure today to welcome you to the inaugural ICCVAM Method Developers Forum. We're really excited about this series of events, and this is the first in a series that will be an iterative event series focusing on different types of toxicities and different endpoints and the development of methods to address those endpoints. So if I could have the slides up, please. I'm going to take just a few minutes to kind of orient you to what we're doing today and what we hope to achieve, a little bit of background, and a little bit of basic housekeeping rules. So as I mentioned, this is the first in the series of method developers forums that we are launching. Today, we are focusing on new approaches for carcinogenicity testing, and we have a really exciting lineup of presentations over the next 2 days. So if you could go to the next slide, please. Just a little bit of background. I think that folks are familiar with NICETM and ICCVAM, but in case you're not, we are the U.S. validation body and the U.S. federal resource for advancing the science of alternatives to animal models and establishing confidence in those models for regulatory use and use in regulated industry. And our function is really to coordinate among all of the federal agencies that either require or consider chemical safety testing data and are interested in moving towards incorporation of human relevant, efficient, and comprehensive methods for improving public health protection. So we keep expanding. As of now, we have 18 agencies that belong to ICCVAM, and those are about evenly divided between regulatory agencies and research agencies. And you can see the ICCVAM family listed on the right-hand side of the screen. And NICETM is an office within the Division of Translational Toxicology at NIEHS, and we provide the scientific and operational support for ICCVAM. Next slide, please. So back in March, we published what I think is really a seminal document, and it was the result of 2 years of hard work by the ICCVAM Validation Work Group, which encompassed almost all of our member agencies. And this is a report on the validation, qualification, and regulatory acceptance of new approach methodologies. And in an effort to operationalize the key concepts that are found in this report, we have launched this series of events, the MDF, or the Method Developers Forum. So this is an effort to try and encourage communication and scientific discourse between method developers and end users of those methods, and to highlight and implement the recommendations detailed within the Validation Workgroup report. And as I mentioned, this is the first, and we anticipate holding probably 2 of these per year, focusing on various endpoints of importance to regulatory decision makers and regulated industry. This is the first iteration, and for this iteration, we are focusing on how methods for carcinogenicity testing align with the key concepts of flexible, fit-for-purpose NAMs validation that are described in this report. So if you haven't read this, I highly encourage you to take a few minutes or a couple hours and read this report. Next slide, please. It is important for us to note that the purpose of this event is to encourage scientific discourse and engagement and dialogue between method developers and end users kind of early in the process to ensure that regulatory needs and needs from our friends in regulated industry are being considered as methods are being developed and optimized. Participation in this event does not in any way imply regulatory acceptance or even regulatory consideration of a method as part of a regulatory decision-making framework. Again, it is intended as a forum for open scientific dialogue. Next slide, please. We have a really exciting agenda over the next 2 days. So we have 10 presentations in total, 5 today and 5 tomorrow. And to ensure that all of you stay fresh and focused and engaged, we'll be having 2 breaks throughout the course of the agenda so that you can get up, stretch your legs, have a bio break, get some tea, coffee, whatever you need to do to ensure that you can give the presentations your full attention. Next slide, please. I want to say a special thank you to the Method Developer Forum Steering Committee and the NICCVAM support team and the NIEHS AV support team, who are ensuring that this event runs smoothly and flawlessly, providing their excellent technical support as they always do. The steering committee in particular has spent many, many hours planning this event, reviewing the high number of submissions that we got, and selecting the submissions that you will hear today. Next slide, please. The housekeeping basics. So I assume most of you have used Zoom before, but the controls are accessible via the toolbar at the bottom of the screen. If you don't see the toolbar, just hover your mouse at the bottom of the screen. We would ask that you all rename yourself in the participants window. So if you could name yourself with your first name, your last name, and then your affiliation, that will help us keep track of the folks that have joined and are participating today. Next slide, please. We will hopefully have time for questions and discussion after each talk. That is really the purpose of this event, is to ensure that there's time for dialogue and scientific discourse. So we would ask, if possible, that you use the raise hand function. So one of the nice things about the Zoom Gov webinar is that we can actually unmute participants, and you can raise your hand and you can actually ask questions of the presenters. And so there's that little hand button that says raise hand. So click on that, please. And then our host, Amber Daniel, who I have to give a special shout out to because she has really been the engine that has kind of driven this train forward over the last couple of years, from the validation work group all the way to implementation of the Method Developers Forum. She is going to help facilitate this event and be in charge of transitions between speakers, and also she's going to call on you if you raise your hand, and she'll let you unmute yourself and ask your question of the speakers. So I see that at least one of our attendees is testing out the raise hand function and has raised their hand. So good job. Okay. And then the last slide, just a notification that this webinar is being recorded. For those of you that may be watching this in the future, we recorded this webinar to facilitate more widespread awareness and distribution. We will make the recording available on the NICCVAM website at the MDF page. Okay, so that's it for me. I understand that there may have been some technical issues in showing the slides to the attendees. Hopefully that will not be the case for our presenters. The slides that I just went through weren't super important for you to be able to see, but the slides for the presentations hopefully will show up for everyone. So I'm going to turn it over to our first presenter now, who is Dr. Tatiana Chairs from the Department of Pathology, Microbiology, and Immunology at the New York Medical College. So Tatiana, please take it away, and hopefully all of the attendees will be able to see your slides. And please let us know as soon as possible if that is not the case.
Sorry, 1 second. I had to unmute myself. Okay, I see a lot of thumbs up. Great. I'm glad that the slides are showing. So first of all, I'm sorry, Nicole, what is this? I would like to first thank the organizers who put in this wonderful forum together. I am very excited to see today's presentations. And of course, thank you so much for giving me the platform to present work on the chicken egg model, the one we've been very passionate about. Well, we'll start with the method description. Historically, the use of chicken egg model has been contributing to a lot of discoveries in the field of human development, including cardiovascular development. And in the more recent years, the chicken egg model, aside from being used for virology, as we all know, and development of vaccines, have also been used for various models of carcinogenesis, immune disorders, and neurologic diseases. So it's a pretty well characterized model with various usages. In our laboratory, we actually started with a turkey egg model where the turkey would be injected in the beginning of the incubation with a chemical of interest. And then very close to their hatching point, which is on day 28, the eggs will be terminated, and we would assess formation of preneoplastic lesions. However, it was hard to justify this model as an alternative to animal because the termination time point was so close to the hatching. Hence, we developed the chicken egg model, which utilizes fertilized chicken egg. We usually purchase the eggs from Charles River, so they are all pretty high quality material. Eggs are all incubated in pretty rigorous conditions at a set temperature of 37 degrees and humidity of 65%. On day 8, we assess viability and development. Have all the eggs been developed? Those that haven't will be excluded. And then we conduct 3 daily injections into the air sac with a compound of interest, vehicle, or a positive control on days 9, 10, and 11. And then 3 hours after the last dose, we terminate the eggs. This time point has been selected not randomly, but rather to allow for the genetic damage to accumulate over the course of 3 days and also not being fastly repaired. Upon termination, we collect the livers. Why livers? Well, first of all, these are pretty large. Even an 11-day-old embryo, those livers reach up to 15 milligrams, so pretty large amount of material for further investigations. Also, liver is the main organ that is responsible for metabolic bioactivation. So it often is a target organ for chemical carcinogens that require bioactivation. And we analyze standard endpoints for genotoxicity, which in our lab are formation of DNA adducts and strand breaks. And optional endpoints include genomics, histopathology. But again, this list can be extended since it is the whole organ. We use various vehicles depending on the properties of the compounds. The most commonly used is cauliflower oil for lipophilic compounds. Quinoline we often use as a positive control. It's been working very well in our laboratory. The dosage selection is based on LD50 in rat, but we also could be limited by the solubility of the compound or toxicity, meaning that the dose that caused more than 50% mortality within the group will be not included in the experiment to avoid any false positive outcomes. We usually analyze at least 2 compounds per experiment, 3 dose levels plus control, and at least 3 biological replicas per group for endpoints. Here's just the types of DNA damage that we assess in our laboratory. I know that other laboratories also assess other endpoints like formation of the micronuclei, but we are focusing more on the direct DNA break, a structural distortion in the forms of bulky DNA adducts, DNA backbone damage, and we can also modify some of the assays to assess oxidative DNA damage. And then you can also see the cross-linking if that does occur. So the two assays that we are usually using for the assay is a 32P nucleotide post-labeling assay, which in which we label a bulky adduct with the radioactive nucleotide, and then through the series of digestion and thin layer chromatography, we visualize the presence of adducts. I must say here that since the eggs are incubated in pretty rigorous conditions, they're not exposed to any other environmental factors, and the control groups and the vehicle control groups, we don't observe any background adducts, unlike with the rodent experiments where there's some background present here. The vehicle controls do not have any background adducts. So if we do see a spot on the TLC chromatogram, we know that it is an adduct and it is due to the effect of the chemical. So it's very easy qualitative assessment of formation of the adducts. And then we can also measure the intensity of that spot later on. And for the DNA strand break, we use COMET assay with the alkaline conditions. We are interested in the single strand break formation. And then we assess the intensity of the DNA in tail. That intensity, if it's reduced, can indicate formation of the cross-links. If it increased, can indicate the formation of a DNA strand break. And then we can also modify the protocol of this assay by adding enzymes that are specific for removing oxidative DNA lesions. Thus, we can assess oxidative DNA damage if we see increase in the DNA strand break after the digestion with these enzymes. We know that the strand break is likely due to the oxidative damage. So the egg model has numerous advantages. This is an intact organism, very complex structure, which resembles in vivo conditions. But since the termination occurs about 10 days before hatching, for the chicken egg, the development is about 21 days. They hatch on day 21. But because we terminated faster, they're not considered to be animals. So we can test pretty large amount of eggs, which means we can analyze a large amount of chemicals or dose levels. And we know that the compound is delivered into the tissues from the air sac. I will show you some of the data to prove that. That air sac, the choriono-allantoic membrane, is lined with choriono-allantoic membrane, which resembles respiratory epithelium. The model is capable of intrinsic metabolic activation and deactivation, so we don't need to use any S9 fraction, and very important, it already has phase 2 metabolic activation as well, which is so important, which is closer resembling in vivo conditions. It is specific pathogen free, so again, we're limiting any potential artifacts that can influence our results, together with a rigorous environmental control. We can evaluate multiple critical endpoints, not just for gene toxicity, but also histopathology, genomic changes. And by that, we're not only given a binary answer of yes, no, but also we can elucidate some of the mechanisms of action. Of course, like any other non-human model, it has limitations as there could be species difference. Again, this is an avian model. It is a developing organism, so there's a fast division of the cells, potentially faster DNA repair processes occurring. While it is a metabolically active model, there are some, of course, metabolic differences between the avian species as well as mammalian species. And the route of exposure, we inject only into the air sac, so it is maybe more difficult to repeat some of the routes of exposure that are present with other exposures. So, and then another thing, we don't know the sex of the eggs, of the fetuses in the egg until we genotype them. However, that did not seem to affect our outcomes that much. We haven't observed too much variability within the groups in our responses. So it doesn't seem like sex plays that much of a role in responding to environmental chemicals. And I will speculate a little bit on why it is so in a couple of slides. So context of use, I don't think that I need to go over the whole process of carcinogenesis, but it's basically just a few steps in which a normal cell under the influence of various factors becomes initiated cells. And then after the series of proliferation cell cycles and clonal expansion, it turns into the preneoplastic foci and eventually into tumor, which can metastasize and invade. And the chemicals play their role at different stages of this process. Some of the chemicals can only induce direct DNA damage. Some do not have that capability, but most can do both. And in order for us to answer the question whether the chemical can affect the normal cell and turn it into the initiated cell or tumorigenic cell, there are several assays that I used. The classic standard battery includes assays for mutagenicity, such as NIEHS assay, as well as plastogenicity and oncogenicity, such as nucleus assay from preparation tests. However, in the recent developments, the use of in vivo tests are getting less and less popular, and in some cases even prohibited, like in the case of cosmetic ingredients. And also we try to minimize the use of in vivo assay where they're not as necessary. However, we know that in vitro test battery can be often over predictive. So for example, with cosmetics, if the cosmetic is positive in the NIEHS assay and in one of the other plastogenicity assays, it might be prevented from further development, which might be detrimental to the consumers and to the industry. So we are suggesting that this chicken egg model can be used as a follow-up for these positive in vitro assays, since it resembles more of in vivo conditions, to in a way rescue some of the chemicals or confirm that the positive genotoxic response is also seen in the model. And then it can be also used for other chemical classes just to minimize the use of in vivo. If it is positive, then is there even a point of going into the in vivo testing? So the method is intended to use for screening, identification of the hazard, and even potency evaluation. We're working on a few publications to address this item. I will not go into this today due to the lack of time, but we can do that as well. It can be used as an in vitro follow-up, minimizing use of in vivo assay, and primarily for cosmetic ingredients, but all other compounds can be assessed in the model as well. And for just now, it has not been used for regulatory submissions, but we're hoping that in the future it will be more open to use this data. Just to show you a few examples of the use of the model, a lot has been published. You can find more evaluations in the model, but this is one of the very good paper because, first of all, it evaluated genotoxic carcinogens, which here in the red, right under the red line, that require different routes of metabolic activation. If we see positive response, we know that the egg model is capable of activating these specific carcinogens. And then we compare them to a less carcinogenic genotoxic or non-carcinogenic genotoxic counterparts, which are chemically structural very similar to these genotoxic carcinogens, but their potential is much lower. And here are some of the outcomes. Here's the COMET assay that measures, again, the DNA strand break. As you see, majority of the genotoxic carcinogens were positive in the assay, again, indicating that the chicken egg is capable to bioactivate these carcinogens. And here's the response with the less or non-genotoxic. Pretty much no positive response with the exception of aflatoxin B2. And here are the results of NPL assay. This is the adduct assay. As you see, the controls, as I was alluding to previously, have no background adducts, so it's pretty clean chromatograms. And here you clearly see the spots. These are indicative of DNA adducts. Now, I know you noticed that diethylnitrosamine did not produce positive response in the model, and that to be expected. Diethylnitrosamine does not produce bulky adducts. which are difficult to detect by NPL assay, that the diethyl nitrosamine addicts are difficult to detect by NPL assay. And that just justifies to use both endpoints, COMET and NPL assay, because in the COMET assay, we had a pretty strong positive response. And here's just an example on how we use that modified COMET assay to confirm the presence of oxidative stress rather than direct strand break. So here's a lefibric acid, which is a non-carcinogenic compound. It was clean in the NPL assay. However, we saw some increase at the higher dosages at the COMET assay. So to clarify whether this increase was to direct DNA reactivity or oxidative stress, we used the enzymes. And as you see clearly with the enzyme digestion, the DNA tail increased drastically, indicating that this strand break is due to the indirect mechanisms rather than direct genotoxicity. Histopathology evaluation is not done routinely, however, it is possible. I included this picture. The paper cited here has way more pictures with the exposures to carcinogens. If you are interested, please check this out. I use this picture more to illustrate the point that the structure, histological structure of the avian liver is very similar to that in mammalian species, including humans. And on day 12, the development is still in progress. You see a lot of spaces that will be filled out by the parenchyma. However, on day 18, that liver is pretty much formed. We also can do immunohistochemistry. Here's an example with pCNA staining to assess other endpoints as well. And this is just a summary table from this paper. Again, I highly encourage you to look into that if you're interested in this endpoint. As you can see, all of those genotoxic carcinogens that I showed before that produced molecular changes also produce severe histopathologic effects. And their counterparts that are less genotoxic, less carcinogenic, did not produce any of the changes. And since the genome of the chicken has been sequenced, we can also use all the next generation sequencing technology that's available to assess the pathways that are involved or even mutagenicity of the compounds. So that's also possible. In terms of the metabolic capacity, I must admit that it hasn't been as well characterized as the rodent liver. However, we know that there are metabolic enzymes present pretty early on in the egg. In fact, day 3 to 4, the egg is capable of metabolic activation. Again, it's an intact system, so they have to work for themselves. They don't have a mother who will conduct the metabolism for them, unlike mammalian species. So we see the inducible activity of phase 1 and phase 2 enzymes. Not only it was inducible, but also comparable to the rodents. This is example with the turkey. In the chicken egg, a PhD student in my lab, Yak Sakar, is working very hard to evaluate some of the metabolic activities in the model. And here are examples with cytochrome, some of the cytochromes, as well as phase 2 metabolic enzymes, and they are inducible by xenobiotics very nicely. And here's the data that I referred to in the past, that male and female have very similar activities in these enzymes, which might explain, at least in some part, why we don't see much variability within the group, despite not knowing the sex of the embryo. And here's more work done by Yaks. It's the profiling of benzoapyrin metabolites in the livers of the chicken embryo fetuses after the injection with a compound. We know that this compound has produced adducts and strand breaks in the model. And now we were able to see also the metabolites present. And from this very complicated picture, which we are hoping to publish very soon, you can tell that the metabolites which are formed and the quantities that are formed are very comparable to the established metabolic pathway of BAP. And here, just to prove that the material that we inject into the air sac is going to the liver, we do not do this routinely. This was just a pilot experiment to see whether we can prove that the material injected into the air sac does get to the organ, target tissue exposure. On the left-hand side are the liver pictures from 2-photon microscopy after injections with the water. You barely see any green color as compared to the right-hand side where the egg was injected with acrid and orange with following our standard protocol, and you clearly see positive staining in the fluorescence in the liver tissues, just proving that the compound did get there. And here's some of the published data. Again, feel free to check out these publications. Some of them are done in the chicken egg, some of them in the turkey egg. Turkey protocol are very similar to the chicken egg. So please look at this if you're interested. Just to how does our data in the egg compares to the standard batteries? Well, with in vitro genotoxicity, not as well as with in vivo data, which we are very happy about because we are trying to use this model as not a replacement, but potentially to reduce the use of in vivo models. And it's much more closely resembles the in vivo models than in vitro. And also for carcinogenicity data, we see close concordance of the results. And some of these false negatives, so to say, in ovo are actually non-genotoxic carcinogens that are also negative in other genotoxicity assays. So with this assay, we're hoping that we can elucidate some of the mechanism of action and contribute to the AOP of carcinogenicity. We have to date evaluated over 80 compounds with all different chemical structures and groups. And the model has been shown pretty high accuracy, sensitivity, specificity compared to the in vivo assays. And so we are hoping that it will potentially replace or reduce the use of in vivo genotoxicity assays used to investigate genotoxic potential of carcinogens. Here are just some technical aspects of the assays. This is the data from the DNA entail for the COMET assay. I did not include NPL assay just because we always have 0 here, so it would be 0. That graph won't be very informative, but HS15 or coliform oil, but we use most often. That's why there's way more data points for this group. Deionized water also works very well for COMET assay, so does twin 20. And then here's our positive control quinoline, which always produce positive results. And then the viability in the control groups are always above 50% as expected. And when we do talk about the adducts formed in ovo closely resemble those formed in other in vivo assays using the same techniques. This is also a nucleotide post-labeling assay. We do not characterize the adducts formed, meaning we do not use the standards to characterize, but we can do that work. And we, in fact, has done that with the alkenyl benzenes. So here's comparison, interspecies comparison between turkey and chicken egg. As you see, these alkenyl benzenes that have structural features that are required for bioactivation to the genotoxic intermediates produce positive responses in the NPL assay in the form of DNA adducts in both chicken and turkey egg assay. And very similar outcomes were observed in the COMET assay. And then with the help of our colleagues in the University of Karlsruhe, we were able to confirm that those DNA adducts observed are actually the alkenyl benzenes adducts. So the protocols allow to avoiding environmental variability. Again, we do not have things like diet or fighting or medium or incubation conditions. They're pretty standard, they're pretty rigorous. We can evaluate quite a lot of compounds, different time points. We can modify the time points. We can select other tissues to evaluate as well. And the results are pretty reproducible among other species. We have not yet done the interlaboratory comparison, but we're definitely open for collaboration. So if you're interested, please let me know. I'll be more than happy to work together. So I hope that in this allocated time, I was able to convince you that the chicken egg model is a good, reliable alternative model for evaluation of chemical-induced genotoxic and related events. It has high sensitivity and specificity for genotoxic compound and non-genotoxic compounds compared to the in vivo data. And this assay allows demonstration of biological consequences of molecular events, and thus elucidating mode of action. And we can also use it for the mechanistic dose response studies. Again, we can analyze quite a few dosage levels in the model, and hopefully this model will be more acceptable alternative to current animal models as they follow up for in vitro genotoxicity assays. I'd like to thank all of my coworkers and colleagues who helped with producing this data and making it possible. And here's my contact information. I thank you for your attention, and please let me know if you have any questions right now.
Thank you so much, Tatiana. That was a very interesting presentation, and thank you so much for sharing that with us. Hello, everyone. My name is Amber Daniel, and I am a contractor supporting NIEHS. I'm going to be helping moderate the Q&A today. But before we get into that, I've seen that unfortunately some of our attendees are still having trouble viewing the slides. I'm very sorry for that technical issue. It seems to be only affecting a few of you still. If you haven't already, I think now that we're kind of between speakers would be maybe a good time to try rebooting your computer. And if that doesn't work, just rest assured that we're still trying to look for other answers to troubleshoot. And also these recordings are going to be available online for any content that you miss, although due to the length, the duration of this webinar, it'll probably be a few weeks before they are posted online. So again, apologies for that. But at this time, we'd like to go ahead and invite the rest of the attendees to ask any questions. For Tatiana, a reminder that you can do that. You can get into the queue by using the raise hand function, and we'll give priority to those questions.
I also, if you don't mind, I see questions in Q&A, the one that were posted during the talk. So I guess if you don't mind, I'll go over. How is exposure to the target organs assessed within the egg model? So we usually do not assess target exposure. For us, the positive outcome where we expect is a evidence that the chemical has reached the liver. As you see, the controls are pretty clear. So whatever responses we see in the model, we know it's due to the chemical itself. And it will be possible with the use of equipment like 2-photon microscopy, for example, which the college where I'm working at right now is trying to obtain. So we're hoping to do some more work in this area. Do we need to qualify the eggs before using them as a test model? I'm not sure what you mean by qualify the eggs. We are getting the eggs from the Charles River, as I mentioned. They're pretty high quality. They are also used, the same eggs that are used for virology, vaccine development studies. That we always assess they're all specific pathogen free. We get the certificate with every delivery of the egg. And we know they're pretty standard weight. We usually get 60 gram plus minus 5 gram eggs. I hope that that answers the question.
Yeah, thank you. We've got a couple hands raised. So up first is John Elliott.
Hey, I'm coming on. Can you guys hear me?
Yes.
Okay, great. A question, and this is kind of similar to the last question. So when you receive the eggs, these are not clonal eggs or anything like that, right? They have some size characterization or whatever, what you were saying, but they're not clonal or anything.
No.
So if you see a fault, so how do you have? What is your feeling about if you see a false negative? Is that due to the egg donor variability, or is that due to the compound actually not? I mean, is that a real negative? How do you know? How do you separate out the donor response to a compound and the compound response?
Right. So great question. So we usually test there are several, right, things. So we use within the group variability, as well as the dose, right? If we see no dose response whatsoever, it's likely to be the true negative in the egg model. We can always repeat the compound. We usually test up to the highest solubility dose, so we do go pretty high. And especially with NPL assay, you know, we see clearly if there is a positive response, we see it right away pretty clearly. So again, we can always repeat the assay, but the biological, I think we have enough of the biological variants, of the biological replicates in our experiment, to say, you know, if we do see negative, it's usually due to the not reactivity in the egg.
So, okay, so you're either, whether it's dose response curve, you're going to use multiple eggs to
Right.
get an answer to come out, and that will presumably cross over the variability in the donor questions. Okay, all right. No, that's great. No, great. Neat, interesting talk. Definitely. Thank you.
Thank you.
Yes. All right. Thank you for the questions. And I see that we have another hand raised. John Gordon, if you don't mind putting your question into the Q&A box, there are a few more there. Please keep those coming. But for now, we're slightly over time, so we're going to go ahead and move on to our next speaker. So at this time, I'd like to invite Dr. Dan Roberts from Toxis Incorporated, who is going to be presenting the Tox Tracker.
Thank you, Amber. Can you hear me okay?
Yes, thank you.
And slides looking okay?
Looks great.
Okay. So I'll jump right in. So thank you for this opportunity. I think this MDF is a great platform to look at, especially NAMs in the context of different disciplines. So today we're looking at Tox Tracker, which I'll discuss briefly, but keep in mind this is already a used genotoxicity assay, but now we got to think about how to maybe repurpose it for the use of detecting carcinogens, which is a much more broad sense of modes of action and, you know, biological key events. So just to orient people, this assay was born from a transcriptomic assessment of actual genotoxic and non-genotoxic carcinogens. So it was looking at any genes that were differentially upregulated or downregulated between those two categories. And it's been identified, again, in the gene tox field, but mostly to look at not only DNA reactive type of genotoxicants, those direct genotoxicants, but also indirect modes of action. The construct is a bacterial artificial chromosome. And of these 6 genes that were selected based on that transcriptomic assessment, they're engineered and packaged in these chromosomes, which of course have normal regulatory features, et cetera. And they were put into stem cells. So it's a stem cell based assay on a murine background. Of the 6 different reporters that I'm showing here, and it's all color coded throughout the talk, but in red we have BSCL2 and this rhodokin gene that are detecting different types of actual genotoxic effects, such as replication inhibition, fork collapse, and things such as double-strand breaks. That's what rhodokin's for. But then we also have 2 cell lines that are looking at oxidative stress, which is usually implicated in, again, that indirect genotoxicity, but also carcinogenicity. And finally, we're looking at cytostasis with a P53, but also ER stress with DDIT3 in the marine background, which is analogous to human CHOP. To give you a purview of what this kind of looks like, here's some dose response curves. And I love the introduction Tatiana gave with kind of a quick slide on GeneTox 101, looking at Ames and micronucleus and other types of assays, because, you know, if you move away from just your DNA reactive kind of classigens here, such as cisplatin, where everything's going to fire, so here the red DNA damaging markers are coming up. If you were to run this in the classical assays, you would see the same phenotype for these 3 different chemicals here. This is just an example. Of course, Taxol is an antigen. Here we have an oxidative stress inducer and an ER perturbator, if you will. But you can see in Tox Tracker, you have different phenotypes. So here in Taxol, you're seeing strand breaks with a lot of cytostasis. We can add cell cycle analysis to then look at angiogenicity in the context of the assay. But here, of course, the DNA damage response is quite low, but we're seeing mostly this chemical is inducing oxidative stress. And as we know, the arsenicals induce all their damages via this kind of pathway. Tunicamycin, of course, if you ran a micronucleus, you will see varying responses, mostly due to cytotoxicity induced by this ER stress pathway, unfolded protein response. So that's kind of why the assay is being used, but what is the assay? So I mentioned again, it's a stem cell-based reporter assay. We start with the dose range finding experiment in wild type cells. So these do not have the GFP reporter. It's a 96-well plate format. We're able to go 20 dilutions wide, so a few logs of coverage. It's a seeding, 24 hours later, an exposure, and then 24 hours after that, we're doing the endpoint analysis by flow cytometry, which will give us not only cell counts, but we're able to look at autofluorescence here at this stage. That way we can deal with those compounds when we do our 3 definitive experiments. The definitive experiments look like this. We have our 6 reporter assays. And again, if it's autofluorescent, we'll add in the wild type cells again. Here we're going 5 concentrations wide, and that's to support benchmark dose analysis. And generally we use 2-fold dilutions. Obviously, if you have a steeper, tighter dose response curve, you can change that up as you need to. It's the same timing. So on day 3, with and without S9, we're doing our endpoint analysis by flow cytometry. And then we're able to look at DNA damage, cytostasis, the oxidative stress, and protein damage all together. But before we start, analyzing data, of course, we want to make sure data are acceptable for analysis. So this endpoint is currently undergoing OECD test guideline creation. We actually owe the WNT a copy, I think, within the next few weeks. So it's currently on my desk for internal review. The positive controls, obviously, step 1 is, do you have a valid assay? Here's an example of some data, and I'm using my mouse, but you can't see it. So let me see if I can get the laser pointer here. But here's a vehicle control. And after you induce with, this is, I think, cisplatin as an example, 2 different doses, yes, 2.5 and 5 micromolar. You're going to induce an increase in gene expression. So this would induce brotikin, BSCL2, as well as the BTG2, which is P53, which you would expect from a genotoxic chemical. So once you know that the cell systems are working, you then, of course, can look at basal background levels of fluorescence. So here's just, for people that like controlled charting and things like that in a regulatory context, you can do that with this. Look at background GFP, MFIs, and this is just an example across 2 different vehicles with and without S9, just to make sure that, voltages are set correctly and the assay is going to perform as you would expect. From there, you can start your calling of the assay. I have more on this later, but essentially we run 3 independent definitive assays, and these are the calling matrices that were used for all the different validations that I'll be talking about soon. But basically a 2-fold response. This line here would indicate a positive result. Anything below 1.5-fold we consider just bounce, and then anything in between, of course, is equivocal. Practicalities, in the interest of time, because I know we're a bit behind, I'm not going to go too much in detail here. But we, in the standard 96-well format, we can do about 25 compounds a week if you're thinking of big library kind of analyses. However, it can be miniaturized if you're looking to look at only a few of the cell reporter systems. So if you do 2, you can bop down to a 384-well format and really increase your throughput. Various solvents are used. We can even use harsher things like acetone. And then a lot of different modalities have been pushed through the assay in the last 10 years. A little bit more on that later, but everything across different sectors, including nanomaterials, polymers, UVCBs, which I saw in the chat earlier. But obviously, most of this has been driven with small molecules because that's where a lot of gene tox work is conducted. Yeah, I got to move this little bar here that you can't see. All right, so context of use. So what is it being used for? I had mentioned, again, it's a gene tox test, which is mostly done for hazard ID, but it can be used and contextualized in terms of risk assessment, getting values, points of departure that you can then kind of look at safety margins, et cetera. It's widely been used to discriminate direct genotoxic effects. So we're looking there at like DNA alkylation, versus indirect effects such as aneugenicity or things that are induced secondarily due to oxidative stress. So edoxo G formation, for example, leading to mutations. Here, when we're thinking about carcinogenicity, I wanted to show that not only, because a lot of work has been done comparing rhodokin and BSCL2, the two DNA damage pathways, to different benchmark dose analyses, different potencies. It's been pushed, those data have been pushed through IVE pathways, but we can also do this with the other reporters, so oxidative stress and protein damage. And the reason that's important, and this is just showing you can potency rank, for data poor compounds to triage testing, and I wanted to skip ahead real quick, is because those are also very important in the context of initiating the process of carcinogenicity. Last year, a paper from LASA came out, and I believe they're presenting tomorrow in this forum on the different pathways that the different biological key events that are important in terms of rodent carcinogens. So over 13,000 chemicals were studied, and as you can see, looking at the top three, DNA reactivity, obviously alkylation, mutation, induction is the first big key event, but then the next top two or top three are oxidative stress and protein reactivity. And, just briefly, these are the pathways that are the focus of Tox Tracker, but the second and third key event buckets, if you will, have not really been as extensively validated as our two DNA damage marker endpoints. So this would be interesting to kind of explore, especially in the context of the chemicals that were used to build this AOP. So yeah, sorry for that segue, but thinking of risk assessment and looking at benchmark dose analyses to find points of departure, you can then easily do some rank prioritization of chemicals. This may allow for data poor chemicals to be higher prioritized for in vivo assays. And one of the things that's important with mode of action assessment, of course, is antigens. And in the context of reporters, we don't have one for antigenicity, but we can focus on the biology there, which of course is spindle assembly checkpoint heavy with that cytostasis short term at 4 hours with the G2M block, and then a 24-hour with the introduction of cells that are in the polyploid state due to mitotic slippage. So that can easily be incorporated into the assay as well. So in thinking of those contexts of use, again, hazard ID, you can either do bare bones 384 well, which I didn't touch upon in detail, but that is being done right now actually in the field. Or what we suggest, of course, is to use the full panel of cell lines because you can classify mode of action in the context of looking at that genotoxic signal. Risk assessment, it's amenable to BMD analysis. You can derive administered equivalent doses after an IVE kind of platform, which seems to be where everything's going, as well as in the context of AOPs, which I'll touch upon later. And those quantitative PODs do overlap with those derived from animal studies, which I'll show in a bit. It's 3 hours aligned, of course, because it's in vitro, but also has high predictivity to in vivo outcomes, tested across modalities. And there are some gaps that I mentioned. Right now we're focused mostly on DNA reactivity, but there's other reporters in the assay, which I think can be used in the CARC field. Putting it all together as well, just focusing on that second bucket, the oxidative kind of carcinogens. I like to think of if the panel is trying to put together some sort of in vitro alternative to give you answers, it's probably going to be a combination of different methods, honestly. I mean, if you just even look at the GeneTox battery, right, we use a combination of tests. And that's by design and for a reason, obviously, to make sure you don't miss anything. But also each assay, no assay can do everything. So here's an example with potassium bromate. Just, you start with Tox Tracker, you see a huge induction of oxidative stress, some cellular cytostasis with a little blip in strand damage. You can do things such as co-treatment with glutathione or N-acetylcysteine to see, okay, what's causal here. Here, if you treat in the presence of N-acetylcysteine, the DNA damage in red here goes away, but oxidative stress still remains, but is knocked down. Basically, this suggests that this large oxidative stress is inducing the DNA damage secondarily. I'm not going to go into the COMET or repair deficient cell lines, but basically, the penultimate here to look for actual mutation would be sequencing the genome, and this assay is amenable to integrating that as well. So here you see a lot of C to A mutations. This is using error-corrected NGS, which we'll hear a lot about tomorrow. But essentially, this points again to high formation of 8-oxo-G. So taken together in the context of even just those three things, you can easily point to oxidative stress as being causal, which is kind of a uniqueness of the assay itself. That's where it started, showing you oxidative stress is inducing those DNA damage kind of responses you're seeing in the other assays. I mentioned AOPs, and here's one on DNA damage leading to chromosomal aberrations. I believe it's the second AOP that has been published in the wiki for gene tox kind of centric events. Rearranging it's still in the same order. Oxidative DNA damage, if you have an adequate repair, can lead to mutations and strand breaks, which of course can be manifested as a chromosomal aberration. And these are just some of the tools in green that you see, many of which are tox trackers that kind of you can use to point and say, hey, this AOP is operable. So this is kind of thinking of potassium bromate, right? Oxidative DNA damage. This AOP is operable, so this must be the mechanism, and it can help you kind of characterize your risk. All right, so that's kind of the introduction. Getting to the meat of it for the last 5-10 minutes here before questions. Thousands of compounds have been tested so far. Most of them have been commercial projects in the last 10 years. But initially out of the gate, it was validated with ToxCast and ICCVAM suggested libraries. And the ICCVAM library was kind of unique at the time in 2016 because it was at the time when we realized how many misleading positives we were having in the mammalian cell gene tox assays. So a lot of that library consisted of chemicals that you should run to make sure that you have a highly specific mammalian cell gene tox assay. And so when we looked at these libraries, what you can see is by itself, ToxTracker, if used, alone, had over 90% accuracy, both in the sensitivity and specificity as well. But you also get the added information of mode of action. You have that oxidative stress, protein damage markers integrated into the panel. Taking it a step further, again, I mentioned the OECD guideline that we're developing to contextualize it for hopefully regulatory, more widespread regulatory use. We had 7 laboratories listed here on the right that were interested in helping with this endeavor. So thank you to those guys. But we did 64 compounds across the laboratories. So each chemical was tested in at least 2 labs. Chemical selection here of these 64 was kind of agnostic towards sector, so it wasn't too heavy in chemical or pharmaceutical spaces. And half of them were considered genotoxic by weight of evidence, which usually means they had, trustworthy in vivo positive gene tox calls. The expert validation team listed below, so a lot of people that know a ton more about gene tox than I do were included in this, and the guideline is currently underway. But looking at data from that endeavor, just as a quick summary, it overall confirmed, that the assay was pretty sensitive, highly specific for detecting in vivo genotoxicants. So really, those weight of evidence, these chemicals are genotoxic, with 83% between lab reproducibility. And this is only looking at rhodokin and BSCL2. And that's kind of highlighted here in these green shades between carcinogens and non-carcinogens. Those with a genotoxic mode of action were able to pick up. The question, I think, still remains, and excuse this coloring, it doesn't really, this 91% specificity doesn't have anything to do with this orange slice of the pie. But in terms of these non-genotoxic carcinogens, can the oxidative stress reporters and UPR reporter actually help us detect those? Well, it's going to depend obviously on the chemical, but that's kind of where we are in terms of carcinogens. I had mentioned, good overlap, and the reason BSCL2 and rhodokin have been used so much in these comparisons is because they look at mutation and clastogenicity respectively, and all the apical endpoints in both in vitro and in rodents, of course, have been looked at with those adverse outcomes. So what we did here, really, this was John Will's And George Johnson has done a lot of this work looking at points of departure in vitro versus points of departure in vivo. And you can see here that they're plotting the 95% confidence intervals here, which is why they look as a line of these points of departure, but are in vitro. So this is for BSCL2, over here is for Rhodokin, actually overlaps quite nicely with those from the rodent micronucleus assay. And these were, of course, the studies that in vivo had adequate dose response data to calculate a relatively decent BMD. So I had mentioned this twofold increase, and sometimes people wonder why is this such a small fold change? And as you develop any assay, any biologist has what I call a hairy eyeball. You can kind of gauge what feels right, what feels like a departure from norm, but that's not usually good enough for anything that's going to be used in the context of human safety and or in a regulatory context. So you can take data here's data from over 100 studies, and what they're looking at is GFP ratios or variability around what those vehicles were, and then what randomness would give you for ratios using a bootstrapping technique, and you can get this nice distribution. And then when you look at the upper bounds of what we would consider normal variation in the assay in GFP ratios, you start to see 1.5 coming out at the 95th percentile and a little under 2 coming out in the 99th percentile. So this kind of quantitatively gives us assurance that anything over a twofold induction is biologically meaningful. Anything below 1.5-fold is bounce, and again, anything in between would be equivocal. Just about to finish up, but in terms of thinking about this as a gene tox assay, one big difference that I think was done was employing a 24-hour co-treatment in the presence of exogenous S9 fraction. So this is liver homogenate that mimics metabolism. Physiologically, it's kind of more of a, I think, exacerbated system, but it's what we use in gene tox for a majority of things. And as you can see here, we do it for 24 hours. That way, not only can you compare minus S9 and plus S9 directly at the end of treatment, but when you're looking at gene expression, you want to keep that biotransformation occurring. You don't want to wash out in recovery because you may lose some signal. So here's just some data showing that it's working. It works well, and this has been in place for quite some time. So just a concluding SWOT for kind of detecting carcinogens. Strengths, it's already qualified for those genotoxic carcinogens. It's high throughput, very simple gating logic, and easily transferred to laboratories across the world, as shown by the inter-laboratory validation. Currently, it's used for hazard ID, but data are supporting quantitative risk assessment processes, which is something you should probably look forward to if you're looking to replace animal testing. Weaknesses, as I mentioned, the other cell lines haven't really been extensively evaluated. So if anything comes out of this, which may or may not happen, it would be great to do that in kind of a collaborative stance. Again, the focus was on genotoxic carcinogens, and the opportunity here is to maybe create a library focusing on those other pathways that are known for rodent carcinogens. With that, here is my contact information. I did my best to catch us up on time, and I'm happy to take some questions.
Thank you so much. Excellent presentation. Yeah, so we'll open it up to the audience for questions. Reminder, you can use the raise hand function and you'll be invited to use your microphone. I'm not seeing any hands up right now, but we do have a question in the Q&A from Allison Harrell. It says, when you conducted between lab transferability studies, were you able to identify contributing factors that could be mitigated to potentially increase between lab reproducibility?
Yes, it was mostly assay acceptability criteria. So in some cases, if a lab generally had an issue, it was due to maybe positive controls being slightly too potent. So they would be too toxic for evaluation, and therefore the trial would be deemed inconclusive. Overall, when data were valid, the mismatches were generally few and far between. So I think that as long as folks get comfortable with the assay and they understand how the controls are working in the assay, those numbers would come up.
Great, thank you. John Elliott.
And I don't want to take anybody else's time on this. Excellent, neat presentation. Quick question. So this is mouse ES cells. These are grown in colonies?
They are grown almost as a 2D monolayer.
2D monolayer. Okay. And so how are they connected together though? Are they colonies? Can you get this is about sample
They are
preparation.
clumpy.
They are clumpy.
They are clumpy. Yeah. Like, when they like stem cells are weird, right? If you don't have enough neighbors, they die. If you have too many neighbors, they stop cycling. So
Right. So the preparation for flow cytometry, which of course needs single cells, the reproducibility and I guess establishing, was it challenging to establish how to get that sample preparation to transfer that to different laboratories and all that kind of stuff?
yeah, fair question. I mean, technically, no. So even though they kind of grow in those clumps, some pipetting with after a trypsinization with some serum present in the buffer dissociates everything fine, and you get a nice single cell suspension. The only thing, the reason it's flow based, I think, is due to that reason, because a lot of people want to use a photometric readout or maybe do high content imaging, but with how they grow, the high content images aren't that great because they're not flat and thin cells like a Hep G2 or something. So really flow is the way to go.
Cool. Great. Thanks.
Yeah, thank you.
Great questions. Thank you so much.
I see there's a question in the chat.
Oh, is there one in the chat? Great. Thank you. Are the cells proliferating during exposures? Question from Steve Ferguson.
Yes, that is one of the reasons we actually like the murine stem cells, is because they are pluripotent. They do cycle well. The cycling time is about 12 hours. We have, thinking about OECD guideline creation, right? That is kind of a pre-requirement before running the assay, is you make sure the cells are cycling. So we have expected kind of targets to hit with proliferation before jumping into the assay itself. Good question. Yeah, you need proliferation to look at gene tox mostly.
Can I ask one more question really quick too? On your reporter assays, do you I kind of saw some positive control, I think, data in there and maybe some negative controls, but are these specific mechanism for the gene that the reporter is reporting on? Like, are you turning on that gene, or are you activating a general tox pathway that happens to be coming in? Is there a way to separate kind of a little more detailed mechanism of action? I don't know, if you add in detergent, for example, you may cause some kind of redox reaction or something like that, and that causes the whole thing to occur. Or do you have very specific chemical that can turn on and off these reporters to ensure that they're working as expected?
Sure. So the controls, yes, there's usually 4 positive controls used in each assay. So cisplatin, for example, doesn't really trigger UPR. Tunicamycin doesn't trigger the DNA damage responses. And then I believe we use diethyl malate, which solely triggers just the oxidative stress responses. The interesting thing is we do have P53 readout, right? And that tends to bounce around, right? Because cytostasis can occur, and that can be triggered by a variety of different initiating events. But the specificity of the markers has been empirically demonstrated. Now, if people have an idea of chemical tests that should be pushed through to really kind of strengthen that argument, I'm all ears, because I think that's only going to help the assay, be trusted, and that's a big part of NAM acceptance, is the trust.
Great, thanks.
Yeah, thank you.
Right, thank you so much for all the questions. I'm not seeing any others in the Q&A box or any hands up right now. We're doing a great job keeping to time. So right now we're going to go for a 10-minute break. If you have any more questions for Dan Roberts. You can put them in the Q&A over the break, and I'm sure he'll be happy to address those in writing. So we'll see you all back at 10:20 for our next presentation. Thank you. Okay, everyone. Thank you for staying with us during the break. At this time, if I can ask the AV to share our next speaker slides, and it is my pleasure to introduce Dr. Christian Strupp of Goen Crop Protection and Dr. Miriam Jacobs of the UK Health Security Agency, who are going to be co-presenting on the validation of cell proliferation as a key event in the assessment of non-genotoxic carcinogenicity. Whenever you're ready.
Thank you very much, Amber. Just a very quick check. Can you hear me well?
Very well.
Perfect. Thank you very much. All right. Thank you very much. My dear co-presenter, Miriam Jacobs, will introduce herself in a few slides when I hand over to her. This is really just to kick us off. We heard two very interesting presentations, mainly focusing on the area of genotoxic carcinogenicity. What we're doing now is move more into the area of non-genotoxic carcinogenicity, and in particular on the role that cell proliferation assessment can play in those. Next slide, please. And the next. Thank you very much. So with regards to the method description, just starting very basic here in biology, carcinogens can of course either be direct genotoxicants, which are mutagens or clustogens that we have just heard quite a lot about, and where we have already quite a confidence in alternative methods, how to assess. And we saw that we are now fine-tuning in the last two presentations. Really great work here. Or of course, carcinogens can lead to tumor formation via non-genotoxic modes of action. The molecular initiating events for non-genotoxic modes of action can, of course, be quite different. However, most of the non-genotoxic carcinogens would induce increased rates of cell proliferation, which then increase the chance of mutation, which leads to the initiated cells in the multi-stage model, which has been presented earlier today, because DNA replication simply is imperfect. OECD has set up a working group on non-genotoxic carcinogen hazard assessment, and the ultimate goal will be to deliver an integrated approach to testing and assessment, an IATA, in which cell proliferation would be a key event. If I could have the next slide. This is quite a busy slide, and I don't want to take you through all the details of it, but it just illustrates what I have been briefly describing before. You have a number of molecular initiating events that can be triggered, which can then lead to a number of key events, but most of them converge at what is circled here in red, at the cell proliferation stage. So we have a kind of a gateway in which we can assess the potential for non-genotoxic carcinogenicity pretty well. At this point in time, I would like to hand over to my dear co-speaker, Miriam. Miriam, do you want to introduce yourself and pick it up from here?
Yes, hello everybody, and thanks very much for this the opportunity to present this work to you today and where we want to go. So on the screen here now, you have, this is actually our IATA that we published in 2020, but we've adapted it for the cell proliferation work. Assuming that you have, your chemical has gone through all the standards mutagenicity and genotoxicity testing and has come out negative, then your chemical needs to be tested according to level 1 in the OECD conceptual framework type of thinking. We're looking at existing information and existing data that you can see in the yellow block at the top. The first part, module 1, is the molecular initiating event, very, very early key events with cell injury by cytotoxicity and a cell injury also via oxidative stress. We heard about oxidative stress being a key event in genotoxicity in Tox Tracker earlier. Well, we will also be able to use Tox Tracker in our IATA, and for that reason, you will see in the blue boxes at the bottom where we've evaluated various tools and assays in relation to cell proliferation. We've also included their Tox Tracker for looking at the oxidative stress biomarkers. In the middle section of the figure, you can see we've got inflammation, immune response, mitogenic signaling, and cellular injury and repair. And we have cell proliferation specific test methods that are used relatively routinely, but they haven't been validated. And that's what Chris is going to talk about further with you as the presentation progresses. And again, in the blue box, we can gather cell proliferation in vivo data from 28-day and 90-day studies. You can see in the broken line box at the bottom. And all of this leads forward into sustained proliferation. And that's really the objective of the talk. The next part, so change in morphology that can lead to a tumor, belongs more for cell transformation. And if we go into the next slide, please. This sort of maps out with our IATA how far we've progressed in our evaluations of assays for the different assay blocks and mechanisms involved. So in green, you can see in the in vitro studies for metabolism, gene transcription, cell death, and the short-term in vivo studies, we have test methods, and many of those are validated for those early parts. But the most critical parts that we need to actually be able to start to make decisions for hazard assessment, and the tools that we need are really in relation to that interface between the in vivo and in vitro cell proliferation space for non-genotox hazard assessment, that lower box in red at the bottom. Then that leads forward into the next evidence-based step that we need, which is for CTAs, which we're not discussing here, but it's very strong part, next part of our IATA. Next slide, please. And with this, I'll hand over back to Chris.
Thank you very much, Miriam. So now going a little bit more into the technical weeds of the methodology. Cell proliferation is not a new assay. As Miriam already mentioned, it's assessed both in vitro and in vivo since a number of years, since the 1980s pretty much. What is lacking, or where we are currently falling short, is the regulatory endorsement. And it really varies substantially. And this is not surprising. If a method has not been going through a formal and proper validation, the trust level between different evaluators and reviewers is different in the methodology. So what we were thinking about in this project is to look at key markers of proliferation. Several have been established and can be addressed, and we thought about validating them all in parallel. So you can either have the incorporation of bromodeoxyuridine, or you can look at the expression of cell cycle specific markers, protein markers like Ki67, or you could look at a genomic signature of markers of proliferation. So here, BRDU and also EDU are artificial nucleotides, which are built into newly synthesized DNA, and then you can detect them. You can either do that by immunohistochemistry and fluorescence detection. You can do that by histopathology on tissue slices. You can do that in cell cultures. Quite a variable technique in here. What you can also do is look at Ki67 as a very specific cell cycle marker, which is upregulated for 4 to 6 hours at the peak of proliferation. So this is quite a time-critical assessment, which provides you quite a real-time picture on this. Can be assessed either at mRNA expression level by PCR or also by immunohistochemistry as a protein expression. Or we could look at genomics markers, which would be a to be defined set of proliferation-linked genes that is upregulated as long as the proliferative stimulus persists. If I could have the next slide. Here just a little bit of a graphical presentation, which I borrowed from the literature on BRDU incorporation, just the unwound DNA, you can see in the dark green the altered bases which are implemented into newly synthesized DNA, and then the classical antibody detection. You will find that the literature from which this graphic is borrowed on the right. And also below, you will find a assessment for EDU, which is an alternative marker working through the same system. If I could have the next slide. This is now Ki67, another marker. Ki-67 now is not on DNA level. That's a protein which is strictly associated with cell proliferation. You see on the right side, there's a very nice graphical presentation here where the depth of redness actually gives an impression on the strength of the expression of Ki-67. So in G0, in the resting phase, the cell is essentially not making any. And then you see as you go through the interface, in particular into the mitotic phase, you see a very strong and sharp upregulation of Ki-67. And as the protein is not very short, very long-lived, you have a detectable response, which is also pretty timely and which you can use for time course assessments. If I could have the next slide. There are principally for the application in vivo in animal species no limitations. The method is usable in all species and also in all organs, which is very important because very often the criticism in assay validation is if they are specific to one single target organ or doesn't work in all species. The in vitro proliferation is in principle also unlimited, but there are a few factors to really take care of. Primary cell cultures need to be able to proliferate if they are used. This is not standardly established for all organs. It is for several. So this assay has been successfully used on liver, kidney, lung, and others for a number of years. But this is just a caveat that not for all organs, there are primary cell cultures available which are able to proliferate. Of course, you have the typical limitation of an in vitro test system if you have solubility barriers in there to bring your substance into the system. And what is important is to focus on true proliferation. So it's not cell number which counts. It must be really DNA de novo synthesis which is in there rather than cell counts. If I could have the next slide. What is the proposal? The proposal would be to formally validate these methods after being for 40, almost 50 years in existence, to bolster the regulatory acceptance here. And the idea would be to go in parallel, both in vivo and in vitro, because here, this is also, I think, an important step that has to be taken with regards to non-genotoxic carcinogenicity, to just make sure that we don't try to jump too far and that regulatory acceptance is left behind. So the idea would be to propose cell proliferation assessment in vivo methods as one project, and another one for the in vitro methods, both in forms of, that's a proposal on the table, of course, only in guidelines which are open and which can be added on as more organs, more tissues, more methods become available. The same set of markers would be used in both methods, which of course means they are also supporting each other in there. And the results would be integrated with other results into an IATA as already described by Miriam. Miriam, would you like to take over here again?
What with the next slide?
The next slide, yes.
Yes. Okay, context of use. Next slide, please. So we already have a good battery of assays and some very nice ones coming forward. Very much looking forward to having Tox Tracker coming to the WNT, for example, for genotoxicants. But the methods that we're proposing here will be a key part of our IATA for the hazard assessment of non-genotoxic carcinogens and applicable across the industrial chemical board. And ultimately, we want to refine international regulatory carcinogen assessment, particularly to replace the rodent cancer bioassay, which I think most groups really are in agreement that it has low predictive power for human relevant non-genotoxic carcinogens. And in practice in Europe, apart from agrochemicals, it is very rarely used. I think in the last 10 years or so, ECHA have only requested it less than a handful of times, less than 5 times. So being able to bring forward the cell proliferation tools as validated test methods would make our IATA concrete. It would help very much support that and the decisions for the next cell transformation assessment leading towards a tumor. It would allow hazard conclusions and points of departure for risk assessment also. And what we want to consider here is going forward with doing the validation work, particularly with respect to agrochemical regulatory submissions. And then it could be potentially applied for industrial chemicals, just because we have a huge amount of data for agrochemicals, and we have a lot of partners that will be able to offer their data for an analysis. And this is all absolutely fundamental for adoption as an OECD test guideline, to provide an exhaustive analysis of the data and make, well, if we go into the next slide, please. Next slide. Okay, so I've missed my discussion point here a little. What I wanted to say in the previous two slides, it really is by including, by taking this work forward and providing that evidence base, that we can make a true transition from the in vivo to the in vitro evidence. And we need the test guideline for international acceptance. So now I should hand over to Christian. Sorry about that.
Thank you very much. If we could go 2 slides further. Yes, one more. Yeah. Thank you very much. Now looking into the biological relevance, here with a little bit of a history lesson, sorry for that. Adverse outcome pathways for non-genotoxic carcinogens have really been postulated since very long, since the 1950s. And here I don't want to go through the whole history of the publications, but it started with publications from Armitage in the 1950s. It went through some key work in the 1970s and 80s, which really broadened or set the foundation stone for the concept. And then the concept was really properly developed, described in the 1980s and 90s. Here, 2 key publications in. And what I have put in for those who are interested is the latest review, which has just come out in Frontiers of Oncology this year. So there's also a reference in here for those who would like to look a little bit into the history of this assay. I cut it a bit short in the interest of time. If we could go to the next slide, please. Could I have that? Yes, thank you very much. On the biological relevance still, as said, the uptake into the regulatory domain is not harmonized across the world. However, there have been some first steps at it. US EPA guideline for carcinogen risk assessment from 2005 already endorsed the concept. And there was a publication coming out in 2015, which also gave some hands-on guidance for the assessors, for pathologists, and so on. So the regulatory world is not naive. Many submissions are containing such data, which is also the reason why we might have a slightly easier job on validating this assay compared to other de novo methodologies where not so much data is already available. As we discussed before, we have recently reviewed the methodology in the context of an IATA, and also there is a publication out on the integration onto the modular approach, which as we described before. If I could go to the next slide, please. Important notes in here, cell proliferation will certainly need to be integrated into an IATA. Not any proliferation that can be picked up is immediately an alert for carcinogenicity. So that is something that has to be worked out in the IATA to make sure that we can distinguish true risk scenarios from adaptive responses in terms of proliferation. Some mechanisms are species specific, and comparative cell proliferation from different species can really help understanding if and where adverse outcome pathways may diverge between different species. Here, one of the beauties is that human is accessible in vitro here, because in principle, primary cell cultures from humans and also cell lines could be used for this assessment once validated. What is important is the total number of proliferation, not just an increase in rate at a specific time point. It is very critical to focus on a specific target cell type. If you have a mixed cell population inside an organ and you would like to look at proliferation, it's not enough to just count any cell that looks like it has just proliferated based on the markers. It is important to distinguish the different cell types. If you do that in vitro and you can clean up your cell preparation to be ideally a monocell type or to be almost free of contamination, then of course it gets even easier if you do it in vitro rather than in vivo. But it is important to keep in mind, if you talk hepatocytes, you talk hepatocytes. You're not talking von Kupffer cells, immune cells, or anything else that may be in there. So it is important that technical aspects are addressed in this validation. It can be used to establish thresholds which are preventive for non-genotoxic carcinogenicity. In principle, they also are a certain kind of a potency hurdle for some of the molecular initiating events that can already be addressed very well. If you have a yes-no response only on certain molecular initiating events, the question, can it actually lead to proliferation, will give us additional ground for decision-making and for informed decision-making in the context of an IATA. I could have the next slide, please. In terms of reference compounds, sorry, the table is a little bit small. Here, what we need is, as in every validation, a balanced set of positive and negative compounds. Ample data is already available on different target organs and modes of action. And here on the right side, you will see in the middle column, there's different target organs which are highlighted. There are prototypical activators on the left side. This list is, of course, not exhaustive in any form, but it gives perhaps a flavor, an impression on how versatile this assay is, how it can be used in different settings here. And of course, what is needed is a thorough review and analysis of the available laboratory data, which is out there. And this will be published as well as in-house data from all collaborators in this proposal here in the course of the validation and perhaps even ahead of that. Thank you very much. If we could go for the next slide. Here, just a little bit of literature. And as you may see, this presentation diverges a little bit from assays which are younger, which are earlier on. Here, there is so much data in the public domain that I only cited key reviews or more recent reviews on different modes of action, pathways, where this data is available. So this is, of course, not an exhaustive list of all which is out there in the literature. It would be very long, but it may again give you a little bit of an idea on what is out there. For the liver nuclear receptor agonists, for CAR-PXR, there's ample literature out there, the same for PPAR-alpha, AHR receptor, and also in general. For the bladder, there's also on local irritants quite a lot of literature out there, recently reviewed as well. Duodenal cytotoxicants as well, and also for mouse lung cytotoxicants. So there is already quite a lot of data that we can work with in context of a potential validation. I could have the next slide. Comparison to existing laboratory animal methods. The in vivo method allows to detect the potential for tumor formation in much shorter studies. That's, of course, time-wise already an advantage over the classical rodent cancer bioassay, which is a lifetime assay. But what is important is also that animals typically don't need to be dosed for a prolonged period of time into a degree of severity, into above maximum tolerated dose or at maximum tolerated dose than in the traditional rodent cancer bioassay. What is, for me, even more interesting is that they would allow a finer titration for a point of departure. For example, if you are agreeing in a regulatory assessment that cell proliferation is the endpoint of interest or the point of infliction towards adversity, then you can do a study with more dose levels, less animals per dose level, but you can do a proper benchmark dose analysis. So your fine titration will be much better than in particular the older rodent cancer bioassays, where you have 3 doses and typically the top dose is lost because it's too high dose, the mid dose random too high, too low, and the low dose, again, uninformative because the dose spacing is too far. This would really be helpful here in this end. And in vitro methods are available for several organs already, and they can really close the gap to humans by using human tissue to assess human relevance of a proliferative response. I think these are all advantages which are important to gain into the regulatory assessment. And with the already available data, it should not be a too hard job to formally validate these assays, which are already out there since a while. If I could have the next slide, please. On the technical characterization, if we could go to the next. How have the sources of variability been evaluated? Well, just listing the main factors contributing to variability. Here I've made a split between in vivo and in vitro. In vivo, it's establishing the time of a peak effect. A proliferative effect can, of course, be transient, and you must, depending on the markers you're looking for, you must be able to capture it. So establishing the time of peak effect is one thing that needs to be done. There are ways to overcome it. For example, if you use BRDU or EDU, you can do a cumulative assessment of all proliferations. If you use Ki-67 or genomic signatures, of course, you might have to be very careful that you capture the right time of proliferative effect. I mentioned already counting only the target cell and a clear differentiation from other cell types in the same organ. Also, there's technical solutions available which are quite straightforward. Tissues with a quick baseline turnover are, of course, different than tissues with low proliferation. If you look at the gastrointestinal tract, which is essentially constantly proliferating to just replace the cells which are being removed by physical stress on it, that's a very different tissue to more dormant, already almost sleepy tissues which do not proliferate unless they are forced to. So this also needs to be taken into account. And for if you use BRDU, EDU, the systemic delivery of an exogenous compound has its own technical difficulties, well understood, well established, but there's the problem of clogging of osmotic mini pumps to be mentioned, which very often leads to a dropout of individual data points. Main factors in vitro, the culture conditions are critical. You must give the cells an environment that allows them to properly proliferate. So media extracellular matrix for hepatocytes are keywords to be taken into account. The fate of the cells before they end up in your tissue culture is very important. The medical history, the way how they were obtained, and if they went through cryopreservation, yes or no. Here, most of the laboratories who are involved in this already have made themselves strict criteria, like a door in a club, on what's allowed in and what is not allowed in. Quality is really important here, and screening needs to be part of that. Establishment of this also would be part of the validation. Then the duration of exposure. Again, this topic of some tissues having a quicker turnaround, where a short-term assay may be more indicative, while others may need a relatively long exposure because of the low background baseline of proliferation. Mentioning again the purity of the primary cell culture, if there's contamination with a quickly proliferating cell type inside a slowly proliferating primary cell culture, that could lead to misinterpretations and needs to be controlled in the validation. I could have the next slide, please. On the robustness, there's ample repeats of studies with prototypical inducers of proliferation already available and out there in the literature, even for cryopreserved primary human hepatocytes. And that's among the most difficult cell types that you can handle. Reproducible responses within and across laboratories and several prototypic inducers of cell proliferation have been assessed in industry. We're talking dozens of studies, and the manuscript is in preparation here. So there will be further clarity coming out in the next few weeks, or hopefully only weeks, if not months. Several laboratories competent in the assay or interested in planning to exchange on and develop a standard operating procedure are already part of this initiative that Miriam and myself are presenting today, which is a young one, by the way. It's only a few weeks old. If I could have the next slide, please. On the closing and the contact here, just recapping, the methods are established and already quite heavily used since the 1990s. Currently, there's a loose network of interested stakeholders arising from the OECD expert group out there. Just listing some members in here. So you will see contract research laboratories, you will see internal labs, you will also see industry, and you will also see regulators in there. Quite a nice balanced group of people who would like to push this assay forward. Formal validation may be organized by a subgroup of the OECD expert group on non-genotoxic carcinogens, and funding is needed for collation and independent statistical evaluation in-house, and literature data and subsequent reliable validation based on ring trials. If pharmaceutical companies and CROs would contribute their data, really quite a lot of this effort could be done retrospectively because the data is already available and just needs a thorough compilation and statistical evaluation. But for this purpose, two phases are proposed here. The first phase is preparatory evidence collation. This is putting all the data which is out there on the table and also trying to get hold of as much as possible. Then based on this to see where there are weaknesses in the coverage of the chemical universe in terms of chemical selection, where might we have to feed some prototypical substances in, where we already have enough. And then also to design the validation based on the observed variability. As mentioned, there's already hundreds of data points with the same prototypical substances available out there, which have been used in vivo and in vitro at pretty much the same concentrations conditions, which can be used to assess variability and then to design the validation exercise in here. Once all of this is together, the proposal would be to move into a more formalized step of validation, which would be ring trials, and finally leading to an OECD test guideline for inclusion into the IATA. I would like to thank you all for giving the time for the presentation. Miriam, is there anything else you would like to say besides thank you to all our listeners?
Well, no, just thank you for listening, and we hope this is of interest.
Thank you.
Thank you both so much. We have a few questions. I'm only going to take one live. See, Lynn Gann has their hand up, so I'm going to prompt you to unmute yourself and ask your question, and then we'll ask any remaining questions be answered in the Q&A box. So, Lynn?
Sorry, I didn't raise my hand.
Okay, no problem.
I think it was the mistake.
Okay, that's okay. We'll go on to Xiaoqing Cheng.
I think it's kind of mistake too. Yeah.
Okay.
Yeah. Sorry.
All right then. Okay. Well, in the interest of time, we'll let you answer that question that's in the Q&A box. So I will just go on and introduce our next speaker, who is Dr. Barbara Parsons from the U.S. Food and Drug Administration National Center for Toxicological Research, who is presenting on the CARCIC method. Barbara?
Hi, everyone. Let me just get my laser pointer up here. The title of my presentation is there we go. The title of my presentation is Clonal Expansion of Cancer Driver Mutants by CARCIC, a biomarker of carcinogenesis. And before I jump into the presentation, I really just want to say how appreciative I am to have this opportunity to tell you about the work and to talk to such a large group of people who are interested in carcinogenicity testing. This first slide presents the rationale for developing clonal expansion of cancer driver gene mutants as a biomarker of future cancer risk. Cancer driver gene mutations are prevalent in normal human and rodent tissues. They occur as a process of endogenous process and DNA replication errors, as was mentioned previously. And clonal expansions of cells carrying cancer driver mutations is considered an obligatory step in cancer development. And clonal expansion of such mutants constitutes a biomarker of effect, meaning an irreversible carcinogenesis related change that temporarily precedes the appearance of histologically observable lesions. To strengthen the human relevance of the CARCIC approach, the sequences that have been selected for interrogation are highly conserved across species, and they include the most prevalent hotspot cancer driver mutations in the 10 deadliest human cancers. And I've listed those cancers there, as well as the genes that are represented in the panel and the hotspot cancer driver mutations in those genes. This slide presents an overview of the approach. The input for the method is DNA that can be isolated from any human, rat, or mouse tissue. The first step in the process is to individually amplify 13 to 15 regions of cancer driver gene sequence using PCR primers that have random bases at their 5' ends. These random bases constitute a unique molecular identifier sequence because all of the products synthesized from a particular parent molecule should have the same UMI and they should have the same sequence. So this use of UMIs allows you to perform error correction by consensus. The products synthesized from the same sample, DNA sample, are pooled, libraries are prepared and sequenced. And then for error correction by consensus, the reads are sorted based on these UMI sequences, and recovery of 2 reads with the same UMI and at least 90% identity across all the reads with the same UMI are the criteria used for mutant or wild type base calling. And in this process, you create single strand, what are called single strand consensus sequences. So the output of the method is error corrected single strand consensus sequence data, which are then used to calculate mutant fraction. And mutant fraction is defined as the number of mutant single strand consensus sequences observed divided by the total number of single strand consensus sequences measured at every sequence position. The Carc-seq approach measures mutant fraction with high throughput. And what we do now is we routinely analyze at least 300,000 single strand consensus sequences for every base of every sample. We only consider mutant fractions that are greater than equal to 10 to the -4 for 2 reasons. First, it's fit for the purpose of quantifying clonally expanded mutants. Larger clones are potentially more important in terms of mutagenic potential than smaller clones. And secondly, using this cutoff eliminates uncorrectable PCR errors that may occur in the first few cycles of PCR before these UMIs are attached at both ends of the molecules. Finally, clonal expansion is quantified as variability in mutant fraction using a metric called median absolute deviation. And I'll explain more about this in a coming slide. But first, I want to say that this slide is included in the deck as well as another one at the end that provides lots of methodological details for those who are really interested. Okay, so why would you look at variability in mutant fraction rather than just mutant fraction, and what is MAD in mutant fraction and why did we pick it? Well, some of our earlier work where we did allele-specific competitive blocker PCR and analyzed cancer driver mutations one mutation at a time, showed what's published in this paper that variation in organ-specific PIK3CA and KRAS mutant levels in normal human tissues correlated with the mutation prevalence in corresponding carcinomas. And that is shown, that's what this figure shows. So our variability in cancer driver mutation mutant fraction correlated with cancer driver mutation impact on tumor development. And we didn't just first look at median absolute deviation in mutant fraction. Our initial work sought to look at different cancer risk factors and also look at try and correlate different metrics with tumor response. And we looked at a variety of different metrics, and MAD and mutant fraction always showed the strongest correlations. So that's why we have proceeded with that metric. This figure here is an illustration, a hypothetical illustration of a way to visualize our data. So you can imagine that on the x-axis, you have all of the amplicons analyzed by Carc-seq arrayed from smallest chromosome number and location to the largest. And then on the y-axis, you have the measured mutant fraction level. And when you see things like this, that's interpreted as clonal expansion. So those are mutations that confer a positive selective advantage, and they the cells proliferate and they accumulate within a tissue. And we tend to see these same mutations recurring over and over in different animals. That's why you see these vertical arrays of mutation. So certainly these are adding largely to the variability that you will see in the mutant fraction measurements. But I would argue that if you are talking about changing the proportion of cells in a tissue as clonal expansion occurs, there has to also be some concomitant diminishment of the representation of cells that are not positively selected. And looking at a metric of variability can also capture that information. And that's one reason why I think variability may be a slightly stronger metric of clonal expansion than just looking at mutant fraction directly. So how do you calculate MAD? Over here, bottom right, I'm showing you that I'm showing the mutant fraction measurements from individual samples. And it's not uncommon that we'll see 20, 50 different mutations in a sample, and we measure the levels as mutant fraction. You can calculate the median of those levels, and then for each sample, and then determine the absolute deviation between that each measured mutant fraction and the median. And now you have a whole list of absolute deviations for every sample. And median absolute deviation is just the median of these absolute deviations, just what it says. It's not intuitive, so that's why I went through that. Strengths of the MAD approach are that it utilizes all available data, and it robustly normalizes impacts of individual large mutant fractions. MAD in mutant fraction can be calculated for treatment groups or for individual animals within treatment groups, as I just described. As we go through, it's important to remember that cancer driver mutations show tissue specificity. And we leverage this tissue specificity as a validation approach. So if we're looking at normal human breasts, for instance, we'll calculate MAD and mutant fraction just from the amplicons known to be mammary-specific drivers. And then we compare that where we should see clonal expansion. And then we compare that to what we see in the remainder of the amplicons, which really should just be the spontaneous background. Okay, we were asked to comment on the applicability domain of the method. So this approach is capable of detecting the effects of both non-genotoxic and genotoxic carcinogens, and I will show you some evidence of both of those. To my knowledge, there's no reason to expect that there are classes of chemicals that would be that are carcinogens and would be inappropriate to assess using a biomarker of clonal expansion or not carcinogens. Interestingly, our preliminary evidence gives hints that exceeding the maximum tolerated dose, as defined by terminal body weight reduction of 10 to 12% relative to controls, may reduce clonal expansion. And this is somewhat comforting because it's consistent with the known effect of body weight reduction on delaying tumor development. How is your method intended to be used? So it's envisioned that Carc-seq assessment of clonal expansion could be employed as part of early drug development, using samples collected from repeat dose preclinical safety studies of 3 to 6 months in duration. The figure at the top right there comes from ICH S1B-R1, and it describes weight of evidence carcinogenicity assessment, specifically how different mechanisms of carcinogenesis could be interpreted, but it also shows under non-clinical approaches how data from emerging technologies could also be incorporated into carcinogenicity assessment. And Carc-seq is such such an emerging technology. It's intended to provide a new information stream for building a weight of evidence carcinogenicity assessment as per ICH S1B R1. This novel genetic analysis could be used by regulatory health authorities to determine the need for 2-year rat cancer bioassays. Also, should clonal expansion of cancer driver mutations be observed, then CARC-seq would provide exposure response information that could contribute to a quantitative assessment of carcinogenic risk. Theoretically, I think assessment of clonal expansion could have value for chemical screening, hazard identification, potency evaluation, and developing adverse outcome pathways. Certainly, identification of clonal expansion of specific driver gene mutations, and I'll show you a couple of examples of that, have the potential to inform mode of action. So practically speaking, how could CARC-seq be applied to general toxicology studies, 3 to 6 months of duration? Well, tissues could be prospectively collected and frozen from such repeat dose studies, and then the tissues to be investigated by CARC-seq could be selected based on histopathology, consideration of pharmacologic target, or simply applied to known tumor-sensitive organs of the particular rodent strain you're looking at. And there is the potential that CARC-seq might be performed using DNA pooled from multiple organs. What regulatory space does your method address? The assay was developed primarily for use in drug development and regulatory review of investigational new drugs. However, clonal expansion of cancer driver mutations as an early event could also be useful to derive an early indication of cancer risk in the development of agrochemicals. And the ability to perform the same DNA-based test in rodents and humans suggests that eventually the approach could be useful in the assessment of environmental or occupational exposures. Unfortunately, CARC-seq has not yet been used in a regulatory submission. Regarding understanding the mechanism, how it supports known mechanistic knowledge of the carcinogenic process. As I mentioned before, clonal expansion of cells carrying cancer driver mutations is considered an obligatory step in carcinogenesis. The biomarker provides a direct readout of clonal expansion of human-relevant driver mutations. Thus, in my view, the biological relevance is a strength of the CARC-seq clonal expansion biomarker approach. I just want to mention, I don't have time to go through it, but there are some statistical approaches we've developed in our work that can be used to distinguish clonal expansion from direct mutagenesis. So what well-characterized and understood compounds were used to assess the scientific validity? So far, only 2 treatment studies have been performed using CARC-seq. One on the chemical lorcaserin, a non-genotoxic carcinogen, and benzobifluoranthene, a genotoxic carcinogen. So let's talk about lorcaserin first. This work was recently published in Toxi, and you can see all the contributors listed there on the author line. But I want to say that this was the brainchild of Todd Boursier, the Division of Pharmacology and Toxicology at the Office of New Drugs. At Center for Drug Evaluation Research. And Todd received some funding from the Office of New Drugs to support this work. As I mentioned, lorcaserin is a non-genotoxic carcinogen. It's also a selective serotonin 2C receptor agonist. It was approved in 2012 by FDA as an adjunct treatment for chronic weight management, but withdrawn in 2020 from the US market due to excess cancer risk identified in a cardiovascular outcome trial. These 2 tables that you see here present the results from the 2-year rat bioassay and also from that cardiovascular outcome trial. And they're summarized by the statement up here that lorcaserin caused an increased incidence in multiple tumor types with increased multiplicity, lethality, and metastatic potential in rats and humans. In the rat bioassay, the strongest carcinogenic signal was seen in the induction of mammary adenocarcinomas and fibroadenomas in female rats. we wanted to replicate this, but look at shorter time points. So we analyzed DNA from 1/4 of the mammary tissue of female Sprague Dawley rats treated with 0, 30, or 100 mg per kg lorcaserin, which are the control mid and high bioassay doses. They were treated daily for 12 or 20 weeks, and then Jennifer Foskey at NCTR analyzed 6 rats per treatment group. The top left here shows you how sort of depicts the mutations that were recovered in one treatment group. The rats that were treated with 100 mg per kg lorcaserin for 24 weeks. What is obvious here is that there is clonal expansion of this one particular mutation, PIK3CA H1047R. It's worth noting that this is the single most prevalent mutation that's reported to occur in human breast cancer. If you look over to the right here, you'll see that PIK3CA H1047R mutant fraction increased with dose and this was significant at 12 weeks and also at 24 weeks. We also saw significant changes in terms of the number of rats carrying the mutation at both time points. We calculated MAD and mutant fraction based just based on this these PIK H1047R mutations. And we showed that here in this figure, the number of the tumor response is shown on the x-axis, and MAD and mutant fraction is shown for these particular mutations is shown on the y-axis, and you can see that there is a highly significant and strong correlation between the two. Next, we calculated MAD and mutant fraction using the mutations for all of the mammary-specific cancer driver mutations that were observed, and correlated that with tumor response, and that is shown here by this red line. It is a strong correlation, but it doesn't reach statistical significance, and partly, I think, because you see this decrease in MAD from 30 to 100 mg per kg lorcaserin. And there's some reason to, in terms of decreased terminal weight, these animals had exceeded the maximum tolerated dose. So that may have contributed to that. The last thing I want to mention about this is that when we calculated MAD from the remainder of the amplicons, not the mammary-specific drivers, you see a much weaker correlation. Moving on to benzobifluoranthine, it's a genotoxic mouse lung carcinogen. The samples for this work came from animals treated at Health Canada by Francesco Marchetti's group there. But generally, this study is being led by Carol Yock and Francesco Marchetti. It's a multi-endpoint international collaboration for which Dr. Yock received Burrow Wellcome's Innovations in Research Science, a Research Science Award. And the study design is shown on the top there. Our part was to perform Carc-seq on DNA isolated from the superior lobe of mutant-mouse males treated for 180 days with 0, 6.25, 12.5, or 25 mg per kg per day using 6 mice per group. Again, the Carc-seq work was conducted by Jennifer Foskey with bioinformatic support from Bin-Shin Gong, also here at NCTR, and the other collaborators are listed there. Here is a visualization of the Carc-seq data. You can see that there's lots of recurrent mutations at particular locations. And then the other thing that's pretty obvious is that all of the highest mutant fractions are red, meaning they come from this 12.5 mg per kg dose group. So we calculated MAD in mutant fraction just from the lung-specific drivers, and that is shown here. There's a significant effect of dose based on ANOVA, and in pairwise comparisons, we saw significant difference between the control and the 12.5 mg per kg. We also saw significant induction of epidermal growth factor receptor mutant fraction, also a major determinant of human lung cancer. And finally, when we calculated MAD and mutant fraction from the non-lung specific drivers, we saw no correlation with dose. A colleague of mine commented that these changes in MAD are modest looking. So I added a little more information here. I am providing the mutation metrics for the animal with the lowest MAD and the highest MAD down here in this particular table. And just because MAD is a measure of central tendency, it doesn't change greatly, but the underlying mutational observations are really fairly substantial. So histopathological analyses were performed on different lobes, a different lobe of lung from the same mice. The results are shown in this table here. And this was performed by Sabrina Ken, Jürgen Funk, and Andreas Seller at Hoffmann La Roche in Basel, Switzerland. Their conclusion was that there was very subtle bronchoalveolar hyperplasia in a few mice that would most likely not have been considered test item related if the mode of action of the compound were not known. There were no tumors in the BBF treatment. One of 3 positive control mice treated with ENU had a lung adenocarcinoma. If you look here on the right, here's the MAD and mutant fraction dose response, again, overlaid with the percentage of mice that demonstrated hyperplasia. And you see the highest effect was for both was in the 12.5 mg per kg dose, and in both there was a decrease in the 25. And again, this may be due to exceeding the maximum tolerated dose, because here I'm showing you the correlation between BBF dose and the weight of the mice. And those 25 mg per kg mice, again, had exceeded the maximum tolerated dose. So how does your method provide information that is equivalent or better than that from existing methods used for regulatory purposes? How does your method contribute to reduction, refinement, or replacement of animal assays? So no test for clonal expansion currently exists, but such a test is needed acutely for the identification of non-genotoxic carcinogens. Assessment of clonal expansion in early drug or agrochemical development pipelines or in chemical evaluations using tissues from less than or equal to 6-month repeat dose rodent studies is intended to generate information to predict tumor response in the absence of bioassay data and without the use of additional animals, because these would be piggybacked on studies already being done. So in this way, it could be considered a refinement of animal use. But negative carc-seq findings in such a circumstance could be included as part of carcinogenicity assessment and ascertain that a rat bioassay is not needed, and then this would reduce animal use. Also, early positive carc-seq findings could result in dropping a particular drug lead and investigating a more successful one. And this would reduce late-stage development failures due to unexpected carcinogenicity. And that translates into unproductive animal use. So technical considerations. How has the robustness of the assay been evaluated? So I told you we correlated MAD and mutant fraction with cancer risk factors, and one of those is human age. That's shown here. So we took normal human breast tissues, calculated MAD and mutant fraction from the breast-specific drivers, and we could demonstrate there was this significant correlation between MAD and mutant fraction and age, which again is a major cancer risk factor. And you don't see that correlation when you calculate it based on the non-mammary specific drivers. And we did the same thing shown right here, but looking at normal human lung tissue DNAs. We also correlated MAD and mutant fraction with historical bioassay control tumor response data. Looking right here, what we did was we calculated MAD and mutant fraction from Fisher 344, Wister, Han, or Sprague-Dawley rats, 10 per treatment group. Well, they weren't treated. They were just 16-week-old rats. But we were able to correlate that with, as I said, historical control bioassay data on spontaneous mammary neoplasms, and the correlation really was perfect. And you don't see it when you calculate MAD and mutant fraction based on the non-mammary specific drivers. And down here, I'm showing you the same thing again, but here we looked at lung tumor drivers, male and female, CD1 and B6C3F1 mice. Again, MAD and mutant fraction was correlated with historical control mouse bioassay tumor responses when MAD was calculated for lung-specific drivers, but not when it's calculated for the remainder of the panel drivers. And finally, how has intralaboratory reproducibility been evaluated? So for the leucocerin work, the samples that were treated for 24 weeks, all the dose groups, they were analyzed in two independent Carc-seq analyses, and those results are shown here. Across the two analyses, 257 mutations were recovered, of the same mutation was recovered in both assays, and this is showing you the correlation between those mutant fraction measurements. And here is the same thing, just showing, focusing on the PIK3CA H1047R mutant fraction measurements. They're highly concordant. And then we also looked comparing Carc-seq measurement of mutant fraction with that obtained using orthogonal methods on the same samples. Still in the leucocerin samples, here we're measuring PIK H1047R by ACB-PCR and showing the mutant fractions are highly concordant with the Carc-seq measurements. The same thing, measuring KRAS and PIK3CA mutations in human breast and breast tumors or human lung and lung tumors. Carc-seq and ACB-PCR mutant fractions are concordant. And lastly, the normal breast tissues or DNAs that were analyzed by Carc-seq were also shared with TwinStrand. And these are the collaborators at TwinStrand who measured mutant fractions by duplex sequencing, and you could see that those two measurements also are highly concordant. So how has transferability been evaluated? Unfortunately, the Carc-seq method has not been reproduced in another lab. I'm certainly open to collaborations in the analysis of samples or helping to set up the performance of Carc-seq elsewhere. As I close, I just want to again acknowledge Jennifer Foskey, Bin-Shen Gong, and Tan Borsier, really for their great assistance in conducting this work and also in preparing this presentation. And thank you so much for your attention. I'll be happy to answer any questions if there's still time.
Fantastic. Thank you so much. We're a little bit over, but I'm going to go ahead and just delay our break and go ahead and have you answer these couple of questions that are in the Q&A. So the first one from David Bill is for BBF, what time points did you get statistically significant clonal expansion?
So we only looked at one time point, different doses, one time point, and it was 180 days. And we're currently working now on the same doses, but animals treated for 90 days. And if we see a signal there, we will go back to 28 days. So for right now, it's just 180 days.
Thank you. And then last question, is CARSeq applicable for assessing off-target insertions for gene and cell therapies that could be driving secondary tumorigenesis in patients?
No, I would say no. Really, Jesse Salk is the person you want to ask that question to. CARSeq does, so duplex sequencing is a better choice for that because it has greater sensitivity and you're trying to find very rare events. CARSeq is more appropriate for looking at clonal expansion where the mutation levels are higher.
Thank you so much. Any other questions for Dr. Parsons? I invite you to please put those in the Q&A box. And now we're going to take a quick 5-minute break and we'll come back for our last presentation at 11:30 Eastern. Okay, welcome back. It is my pleasure now to introduce our last speakers, a co-presentation by Dr. Leslie Riccio and Dr. Jamie Scaglione, both from Cytovation, who are going to be speaking about human relevant genetic toxicology for risk assessment.
All right, thank you. Can you get the next slide? I think you need someone to advance my slides for me. Can you advance slide? Okay. So I'll be talking about a human relevant genetic toxicology, as the title said. And this has been a collaborative effort over the few years, 6, 7, 8 years, between my lab at previous company I worked for, and more recently in my current lab at Cytovation. The collaborators are Bevan Engelward from MIT, who developed a DNA damage assay that it's called Comet Chip. It's basically the equivalent of the Comet assay, except it's a high throughput format, running 96 well format, and with automatic imaging and quantification of the comets. This has been funded through the NIEHS SBIR grant program. Thank you very much, NIEHS. We also have been collaborating with Carol Yawk for a number of years. She, when she was at Health Canada, and also now she's at the University of Ottawa. And this has been funding some of the other efforts from using the micronucleus assay and the mRNA biomarker called TGX-DDI, which is a biomarker was used to identify genotoxic mode of action in human TK6 cells, HepRG cells, and a number of other cell lines presently. It's also being employed by the EPA high throughput testing program. Next slide. So I'll be talking about as a new approach methodology that's focused on key characteristics of human carcinogens, genotoxicity, and mutagenicity. And that first, as a reminder to folks, this is what the regulatory genetic toxicology test battery consists of. It consists of a test for gene mutation in bacteria, a test for chromosome aberrations or micronucleus assay, which is equivalent, and an in vitro mouse lymphoma assay, which is a gene mutation assay. The follow-ups to these, or the second tier, so to speak, in vivo testing, is again the micronucleus assay, which is typically used in rodent hematopoietic cells, typically, and an in vivo test looking at a second tissue assessment, and this is typically common assay in liver. This genetic toxicology test battery, which was developed basically in 1997, really hasn't changed much. Its focus is a hazard identification focus. It's used to assess potential of a test compound to cause mutation and genotoxicity in a 3-tiered approach. However, these test systems have certain issues that in this day and age where human cells are becoming, you can basically measure all the same exact endpoints in human cells. You're talking about DNA repair deficient bacterial cells. They are excision repair deficient, which makes them very highly sensitive to mutagenic mode of action. Also, the cell lines that are being used are p53 deficient cell lines. In the case of mouse lymphoma cells, it's derived from an inbred mouse cell line that's p53 defective, which is the primary response mode for genotoxic exposures. The in vitro assays use highly induced rat liver S9 to provide phase 1 metabolism only. It's highly induced with phenobarbital and beta-natriflavone, and it's not human relevant. It's a rat that's been highly induced and without phase 2 enzyme conjugation. So you have a disconnect here with this use of S9. The other point is that there's no human gene mutation is required. You have a rodent gene mutation assay, and again, inbred cell line is deficient in P53. The tier 3 is animal testing for DNA damage, chromosomal mutation, and nowadays there's a gene mutation, which primarily has been done in transgenic mouse models. One of the deficiencies of parasite-based NAM has limited application to volatile compounds, but many of the same exact endpoints that I'll be talking about in this presentation are also being developed in respiratory tract epithelial cells, and basically can make the same measurement in those types of cells. Next slide. So what I'm talking about is multiple endpoint genotoxicity immunogenicity assessment in metabolically competent human liver cell line HEPA-RG. And this HEPA-RG cell line is very versatile. And we have published a number of papers using, for instance, the micronucleus assay, which works very well to assess chromosome damage using flow cytometry in a 96-well plate format. This is an assay that's been developed by Litron Technologies over the years, and it's readily applicable, basically seamlessly application to the HEPA-RG cell line grown in either 2D mode or in 3D mode or as spheroids. We also have established the COMET assay to measure DNA damage using COMET chip technology. Although there is an in vivo guideline for the COMET assay, there is no in vitro COMET assay guidance. Perhaps the more interesting ones that may be more applicable across all models is the transcriptomics-based approach. Today I'll be focusing on the TGF-DDI biomarker, which is an mRNA biomarker. This is primarily using what's referred to as BioSpider technology, which is a kind of a high throughput gene expression assay developed by a company called BioSpider, and it's being employed by the US EPA, for instance, as part of the high throughput assay program. And finally, the far side there, error-corrected next-gen sequencing. This is a technological leap that people have been waiting for, at least myself. This error-corrected sequencing was developed by Jesse Salk, who will be talking tomorrow. And this allows a direct measure of mutation and mutational spectra. In other words, the types of mutations that can occur. So all these endpoints can be measured in human HEPA-RG cells. Now, there is a timing difference. For instance, the COMET assay and the transcriptomics can be done basically at the same time point. The micronucleus assay requires cell division. So you have to induce cells to go into division. And the error-corrected next-gen sequencing, again, requires cell division, so 2 rounds. But these are complementary genotoxicity assessments, and you need this to cover the broad range mode of action that genotoxic chemicals can act through. These multiple endpoints are required to make this measurement for both genotoxic and non-genotoxic carcinogens. And as importantly, this allows multiple endpoints to benchmark dose and point of departure when we want to use this types of data for in vitro to in vivo extrapolation modeling. And there's been a recent paper on agrochemicals from Corteva using HEPA-RG cells to look at non-genotoxic mode of action. Next slide. So the context of use is that there's hazard identification, genotoxicity screening, and rodent cell lines are basically are not relevant to humans in the sense that, for instance, one of the reasons people use Chinese hamster cells is that they have large chromosomes. That's one of the basic reasons. If you look at chromosome aberration in vitro data, that is the basic reason that those are used, and they grow well in culture, not really relevant to cancer. And also, the more important thing is to replace reliance on the rat liver S9 based metabolism, because these genetic toxicology assays that are used, the Chinese hamsters, inbred mouse cell line, stem cells from mice, they all require the addition of metabolism because they're devoid of metabolism. So are many of the cell lines that were used in the high throughput testing program. So the use of this highly induced rat liver S9 with no phase 2 metabolism is not relevant to in vivo metabolism of both endogenous biochemicals like testosterone or some of the other conjugates that are from endogenous chemicals and xenobiotics. This is uncoupled, incomplete biotransformation. We don't have phase 2 metabolism. That's illustrated in a case study we published recently. On the bottom panel there, we look at TK6 cells. This is an assay that we do in my group routinely with and without S9. This is naphthalene being examined in the traditional TK6 cell micronucleus assay with S9. During that time period when this assay was first done, my colleague, we were looking at simultaneously a rat, mouse, and human hepatocyte cytotoxicity from naphthalene. And there was a large disconnect what we were seeing with S9 with respect to concentration. As you can see, 10 micromolar, 20 micromolar reduced survival and caused micronuclei. In the case of hepatocytes, rat hepatocytes, primary human hepatocytes, the LD50, cytotoxicity 50%, was about 500 micromolar. So there was a 5-10-fold difference. Later, after we had public funding from the NIEHS on the SBR grants, it's one of the compounds we tested among several. And this naphthalene, we tested it both in the micronucleus assay and the HepRG comet chip assay. And as you can tell, there is absolutely no increase in either the micronucleus assay or the comet chip assay using naphthalene when you have phase 2 metabolism. That's the difference here. You have basically negative response as a follow-up to a positive response in vitro because there is no phase 2 metabolism in S9. Next slide. So HepRG is a versatile cell line. It's been adapted to several formats, 2D, 3D. There's some organoid cultures, and it has been traditionally used to look at things like cytotoxicity, enzyme induction, transcriptomics. And what I'm referring to here today is the use of this particular cell line as a medium throughput genotoxicity assay. It has versatile applications across the board, enzyme induction, chemical screening. It's part of the NIEHS Tox 21 and US EPA ToxCast programs. A lot of that's been led by Steve Ferguson. It's also been used for enzyme induction studies in certain areas of the world where getting primary human hepatocytes may be tough. There was ICCVAM validation many years ago conducted demonstrating that the HepRG cell line is a suitable replacement for primary human hepatocytes to assess enzyme induction studies as part of drug-drug interaction. Next slide. So this HepRG assay is aimed at shifting genotoxicity assessments away from rodent-based assays, because it's kind of a knee-jerk. And there's also companies that or industries that cannot use animals, and there needs to be some type of follow-up for that can be used. And this assay, what we have developed is quantification of regulatory genetic toxicology endpoints that are used in regulatory assays into the HepRG cells. And this allows human relevant alternative to in vivo genotoxicity assessments, particularly when you can't do this. So the assays that are regulatory in nature have OEC test guidelines behind them, are the in vitro and in vivo micronucleus assay. This is a measure of chromosome aberrations. As I mentioned, the flow-based assay developed by Litron can be seamlessly used with the HepRG cells. The only trick that we need to do, this micronucleus assay requires cell division. But that is easily accomplished by adding epidermal growth factor to the cells, and that pushes these cells to go into division and allow the quantification of the micronucleus assay in the single cell suspensions. So there's an in vitro and in vivo micronucleus assay at my group here at Cytovation. We've qualified TK6 cells with and without S9 and HepRG micronucleus assay, and it's ready to be available for testing basically today. The in vivo common assays is OEC 489. It's typically done in liver. It's a measure of DNA damage, detects a broad range of lesions, and can detect DNA repair. There is no OECD guidance. However, there is a high throughput comet assay that was developed at MIT, which allows automated comet assay scoring. And this is in contrast to the in vivo common assay, which is one click at a time scoring. Some technical person is going to sit there hours on end, clicking on one cell at a time to accumulate enough cells to be able to score a common assay to meet regulatory guidelines. In this case, you use a 96-well format in a movable stand with an imager on it, and you can collect these same data in an automated fashion and in a 96-well format. So very high throughput as compared to the common assay that's being done to meet regulatory test guidelines in vivo. The next slide is on the other two endpoints of transcriptome. Transcriptome-based mode of action is becoming the kind of gold standard right now. It's understanding mode of action. It's being used by the EPA and others to assess mode of action. One of the earlier applications of HepRG cells has been to look at non-genotoxic mode of action. And one of the important things that distinguishes HepRG cells from, for instance, HepG2 cells is HepRG cells have a CAR receptor that's expressed and it's active, so you can detect nuclear transcriptional activators, which some of them are known to be non-genotoxic carcinogens. And secondly, the genotoxic mode of action can be assessed using mRNA profiling, TGX, TDI. There's also another biomarker that's mRNA-based called GenoMark. And a recent paper by Carol Yock in the GenoMark labs have shown that these are very complementary and they're basically 100% accurate between themselves. The HepRG error-corrected next-generation sequencing is something that it's ongoing now. We have a small business innovative research grant to develop this. And this is designed to replace 50-year-old clonal selection gene mutation assays. One of the reasons that there's no human gene mutation assay to be done rather than the mouse is that the human cell line requires a 7-day growth, 7-day maintenance of the cells, and no one wants to come in on the weekends every time. So you have to, this is in contrast to the mouse lymphoma cells, which divide every 8 hours, and that can be done in 1 week. So it's a kind of an inconvenience thing. There's no real follow-up to a bacterial mutation assay outside of the inbred mouse cell line mouse lymphoma assay. And finally, to measure gene mutation in liver has required transgenic rodent models. And these rodent models have had limited availability. And nowadays, at least last I checked, it was up to a 1-year lead time and a high cost of more than $200K. With the development of error-corrected sequencing, this by Jesse Salk and the folks at TwinStrand, this allows a direct examination of mutation without clonal assays. And you can do this by allowing cells to divide. And secondly, it can be directly measured in vivo. So again, you have the ability to go in vivo. Same with the transcriptome. One thing I did mention with transcriptome, you can go between in vitro and vitro. And as a matter of fact, the transcriptome, you can go back to historical tissues, formalin-fixed paraffin-embedded tissues, and look at the same endpoint. Finally, there's ability with these HepRG cells to do quantitative dose response analysis. There's been a couple of papers I just talked about where gene tox is moving to benchmark dose, populating some data in outcome adverse pathways, and using benchmark dose to actually look at potency between sets of compounds in the same class, and that enables you to identify the bad actors. You can identify point of departure with using transcriptome-based dose response, and in vitro and in vivo extrapolation modeling is a very hot topic right now. And one of the papers I just mentioned, agrochemicals, done by the Corteva group, uses IVE data from HepRG cells to do risk assessment. Next slide. So with this context of use, we're measuring three key characteristics of carcinogens: metabolism to electrophilic agent, genotoxicity, immunogenicity, can alter DNA repair. And this is directly relevant to the carcinogenesis process because we know that chromosomal aberrations and gene mutations contribute to the development of cancer, adverse outcome pathways, and it also allows to inform cancer risk assessments. And we have done several papers, we as myself, Carol Yock, and Bevan Engelward, and this is a couple shown there on the next slide. So the context of use is we have identical endpoints that are as part of the regulatory bioassay. We can measure both the micronucleus and comment, transcriptomics, and these are all complementary endpoints. So this human multiple endpoint genotoxicity testing platform can be used as a follow-up for positive compounds in the hazard identification regulatory genetic toxicology test battery. It's across multiple sectors because a lot of folks need to conduct the genetic toxicology test battery, the whole thing from in vitro to in vivo, and some labs or some companies are restricted on their use of animals. So this gives you an alternative to animals that have intact phase 1 and phase 2 metabolism, which is a critical factor in determining drug activity or toxicity in vitro and in vivo. Next slide. So this biological relevance, human relevant metabolism, phenobarbital beta naphtha-flavone, rat liver S9 with P450 cofactors only has been the gold standard. However, it's not a replacement for human metabolism. It never will be. This method identifies as regulatory assays, integration of transcriptomics-based mode of action, and again, complementary assessment of multiple endpoints in the same cell line enables this cross comparison and, for instance, identification of benchmark dose. This complementary endpoint is very important. For instance, benzene, a known human carcinogen, and ethylene oxide in vivo, they're more on the chromosomal damage side of the scheme of things. So they have very little induction of point mutations. However, they're really good at chromosome aberrations, and there's compounds that are vice versa. So you have to be able to capture the big changes, chromosomal damage, and gene mutations in the same system to allow the comparison of these endpoints, because one may give you a benchmark dose that's very low, chromosome aberrations may be coming up before you see a gene mutation with respect to dose, and you won't capture that if you're only focusing on one endpoint and disregarding other modes of action. Next slide. So again, continue with the biological relevance. Liver is the most common target organ, and in NTP cancer bioassays, we're trying to replace hazard identification assays with a human relevant assay that allows us to be able to use IVE to make risk assessment calculations. Although we have done data set in the regulatory bioassays that are done, they are typically 3 doses in a control. Mammalian cell assays run like that, and as was mentioned earlier, and so are the animal studies. And a lot of times, the top dose may be MTD or maybe highly cytotoxic, and you get a signal. And so using 3 doses in a control, there's a low confidence benchmark dose calculations that are being done. And with these assays, you can actually repeat the experiment. You can do triplet, you can do 8 doses, 12 doses, and you really can define the shape of the dose response curve that you need to do for risk assessment and to come up with points of departure and in vitro to in vivo extrapolation modeling. Next slide. So technical characterization, we have the funding through SBIR application. Let us bring people on site to train them, and that offer is still available. If there are folks online that want to be trained in the conduct of the use of HepRG cells to make these measurements, you're welcome. Just please contact me. We provided, to minimize variability, we provided cells from the same vendor. They have can basically have proprietary media, and the cells, the compounds were selected and rangefinders were done by my group, and these collaborators conducted these assays. The data have been compiled and undergoing a lot of assessments and discussions. Well, one thing has become clear when you have these screaming positives, they are highly reproducible between labs. When you start getting into some of these compounds that are iffy, you get iffy data in HepRG cells as well, and you try to resolve that by being able to look at multiple endpoints. And I think this is one of the dangers we have is we need to be able to make these multiple endpoints, and not only for HepRG cells, genotoxic compounds, that can also have been used for many years now to look at non-genotoxic mode of action. The micronucleus assay and the transcriptome biomarker have been qualified by 5 genotoxic compounds versus 5 non-genotoxic. Carol Yock has, again, they published another paper using what I refer to as GenoMark. And the HepRG micronucleus assay and the TGX-DDI biomarker have been incorporated into the Health Canada testing program. And there are many, many papers. I looked before I got on the program here with something like 500 something publications or more now with HepRG cells, and it's been a continual increase over time. Next slide. Next slide. Hello. All right. So to end up, I think we're talking about next generation genotoxicity assessments. Haven't talked about the computational tools that are becoming available, not only QSAR tools, but other tools that being used to compare compounds, for instance, using artificial intelligence mechanisms. The computational QSA structural alert probe have been working very well for bacterial to predict bacterial mutagenicity testing. They're so good at predicting bacterial mutagenicity. If you have a contaminant that's positive in these QSAR models, you don't really have to do an Ames. You have to do an Ames test, but if it's negative, you don't have to do an Ames test. And that gives a leeway there. That's how powerful they are at calling something a negative. However, they're not as good at predicting mammalian cell responses. So I think for those, we're still in experimental mode and will be for a while. In our case at Cytovation, we have developed a micronucleus assay, both with human TK6 cells and HepRG cells, the TGX-DDI biomarker, and we have several computational toxicologists that have expertise in doing in vitro to in vivo extrapolation monitoring. So we can move to human relevant genotoxicity assessments using human relevant cell lines and in vitro to in vivo extrapolations modeling to make that risk determination for humans. So with that, these are the contact information. Jamie runs the lab at Cytovation. I'm the CSO there. And if any questions, I'd be happy to answer any questions. And the offer to come train is out of my group is also real. So contact me if you're interested. With that, I'll take questions. And thank you very much for listening.
Great. Thank you so much, Les. I do see one question or two questions in the Q&A box. Can you ballpark HepRG price point?
Well, for the micronucleus assay, it's like comparable to doing the TK6L micronucleus assay, because we're not doing with and without S9 and 24-hour with S9. So we're only looking at, so as far as kit goes, you're using only one kit basically, and the technical person's time is hands-on time is probably less because you're not going through the plus minus S9 rigmarole and the minus S9. So it would be comparable to a standard micronucleus assay. And go ahead, want to read the question, Amber?
No, I was just going to read the next question, but if you wanted to elaborate on that more, go ahead.
Well, I was going to say, on the other hand, error-corrected sequencing is pricier to do. The transcriptomics biomarker is relatively inexpensive. BioSpider, I think, charges $15,000 to $20,000 per plate, 96 well data points, something like that. So it's not out of sight expensive. And as I mentioned, the micronucleus assay is comparable to what we would do with TK6.
Thanks. Next question is from Ming Heng Shaw. How did you measure micronucleus assay in HepRG?
Well, we used a flow-based micronucleus assay, and since these are 2D, they're 2D format. The data I talked about, it's in the publication. And basically, what that flow-based assay is measuring is actually nuclei and small nuclei. So basically, you lyse the cells and use a probe to identify the nuclei. But it's using the Litron micronucleus assay and multi-flow assay. Some of them are on the phone, and so on the call, so we'd be happy to help you out. We got either Steve Derdinger or. All right.
All right, and we have one more question from Steve Ferguson.
Yeah, I just wanted to clarify, I inadvertently moved a question from Lynn Gahn to the answered, and I actually posted a reference, but the question was, how's heparrhs metabolic capacity compared to human primary hepatocytes and or rodents in vivo? And so I just posted in that response in the answered part, and as also just sent in the chat a reference we put out a number of years ago that did a systematic characterization of a number of enzymes. And so that should be a good place to start.
Thank you.
I should have mentioned Steve Bryce as well. He's been the lead person on the in vitro micronucleus assay at Litron.
Well, we are at time, and I'm not seeing any other questions right now. So I would like to say personally, thank you to all the attendees for your interest and engagement, and a special thank you to all of today's speakers for participating. And now I'm going to turn it over to Nicole Kleinsteuer for closing remarks.
Okay, thank you all so much. Just echoing Amber's thanks. Really outstanding presentations and engagement from the audience. We had anywhere between 115 up to 150 plus people online throughout the course of the morning, holding steady at over 100. So really robust participation. And as was noted, we will make this recording available online for folks who were not able to attend today. And really look forward to seeing you all back tomorrow at 9 a.m. Eastern time for day 2 of the ICCVAM MDF focused on new approach methods for carcinogenicity testing. Thanks so much, everyone. Enjoy the rest of your day.